Welcome back to our Network Neuroscience education workshop. While in the morning we focused on mapping structural and functional networks, the afternoon session will delve into a bit more detail about analysing, particularly using graph-based analyses of both functional and structural networks. Our first talk this afternoon is by Bratislav Misis, and Bratislav will consider communication in brain networks. So, Bratislav.
All right, so thanks to those of you who decided to come back after lunch. So our jumping off point is this idea that if you followed all the advice that you heard earlier in the day, you can reconstruct a network of white matter connections between different parts of the brain. And what you have there is not just a pretty picture, but really a map of how any one area could potentially send a signal and influence other areas in the network. So I'll talk today from the working under the assumption that basically this network, this physical infrastructure, supports a myriad of elementary signaling events and processes, so electrical impulses being transmitted from node to node. And the superposition of these signaling events ultimately manifests as patterns of interregional covariance, so functional connectivity. And what I really want to talk about over the next half hour or so is what is the nature of this communication process. And as I go through kind of how different people have thought about this problem, I hope that you keep in mind kind of these three things. When we talk about different communication processes, it's important to think about what's biologically plausible and what's biologically implausible. What is actually suited to the particular and unique architecture that the brain possesses. And finally, the nice thing about this particular set of questions is that all of these communication mechanisms actually make predictions about functional interactions. So what kind of communication mechanisms are actually empirically supported? So one way that you can kind of make sense of various communication mechanisms that have been proposed is to place them on this continuum, running basically all the way from very centralized routing-like mechanisms where you have a specific source and a target and you take the optimal route to get to them, all the way to completely decentralized mechanisms that are more akin to random walks, for instance, where a signal emanates from some node but randomly chooses where to go. And interposed between these two extremes are then a variety of other mechanisms that we can envision. But I'll start out with shortest path, what I'll call routing, which has been the kind of the dominant way of thinking, and for good reason. So if you were to think about just a priori, what would be a good communication model in the brain? One very important set of considerations would be what is the medium of information exchange in the brain? And it really has to do with electrical impulses that are transmitted as action potentials via axons, and then from cell to cell going via synapses. So transmission along these electrical synapses is going to be inherently slow. It's a physiological process, so it's going to be inherently noisy and subject to interference. And it's going to be it's going to be costly in terms of the energy and the material that you need to, for instance, release vesicles, package them up, and so on. So a priori, you might say that trying to minimize the number of synapses that you use would be a really good and desirable outcome. And a good candidate then would be to use shortest paths to communicate. So when we talk about shortest path, it's important to keep one distinction in mind, and that is that between walks and paths. So with a walk, I have a source node and a target node here denoted in red. And by the way, everything that I talk about at the bottom, I guess from your perspective, left-hand corner, I've noted just the various functions that you can use in the brain connectivity toolbox to actually run some of these routines. So you have a source and a target, and basically with a walk, it's just any contiguous set of edges from a source to a target. So it could be something like this. And the length of this particular walk is 5. There's 5 steps. A path, by contrast, running from the same between the same set of nodes could be something like this. So the path length between the source and target is length 3. And this concept, the path length between two areas, tells us how topologically close they are to one another, and importantly, how easy it would be for them to exchange signals, to exchange information. And once you can compute paths in the network, you can actually ask a lot of very important biologically meaningful questions. You can ask, for instance, for a given node, what is its mean path length to the rest of the network? Because if a node can be reached very easily and can reach other nodes very easily, if it has a high closeness centrality, then it's well positioned to distribute information, to broadcast information, and also to integrate information. You can also ask, if I take all possible pairs of nodes and I compute the shortest path between them, what proportion of these paths go through a particular node? So what is its betweenness centrality? If a node sits at the intersection of many different paths, then it's well positioned to act as a bridge or as a way station for signal traffic. It's well positioned to both absorb information from multiple parts of the network, but also to disseminate that information. And then you can take this further and think about the network as a whole. So you can ask, what is the mean path length between all possible pairs of nodes, the so-called characteristic path length? Or it's not exactly its inverse, but close enough, the global efficiency. And the idea is, if a network has a very short average path length, that means that on average, any pair of nodes could exchange signals very easily. So it's indicative of a greater communication capacity and potentially lower signal attenuation. So there's really a very large literature at this point that has shown quite convincingly, I think, that shorter path length is really associated with things like greater cognitive capacity, greater cognitive engagement, normal neurodevelopment, and deviations from this. So increases in path length are typically indicative of some form of pathology or disease. So what's the evidence that shortest path communication is actually relevant or meaningful in the brain? There are kind of two things that we can think of. One is that the brain is a canonical example of a small world network. So a class of networks that actually has path length that's close to the theoretical minimum that you have in random networks. So right off the bat, brains have a close to optimal path length. The other kind of set of evidence comes from what are called minimally rewired networks. So these are networks where the authors took brains from model organisms, so in this case the macaque and C. elegans. They took the nodes and they placed them in their original biologically realistic spatial locations, and they said, okay, I'm going to try and put connections between pairs of nodes such that I minimize the length of these connections. I minimize the wiring cost of these connections. And so when you do that, when you make these minimally rewired networks that are shown in red, they show you can actually drastically reduce the wiring cost in the network. So you can make improvements. But the price of that is that the path length in these networks increases. So having a short path length appears to be a costly feature, but it's one that, it's a cost that appears to be readily incurred, and it might be a property that's selected for in some way. So these are all kind of, I think, good reasons to consider shortest path as a viable mechanism. Shortest path routing is a viable mechanism, but I just want to say just a couple of words about why we might want to think about going beyond just the shortest paths. One is that, so first of all, when you assume shortest path routing, you're actually making two very important assumptions. One is that the network elements, so for instance, the neurons or the populations of neurons, actually know the global topology. They know what the network looks like, and they can actually compute the shortest path from the source to the target, which is biologically implausible. The other assumption that you're making is this. So this is a typical network that you might get from diffusion-weighted imaging. These are the connections in that network that participate in at least one shortest path. So what you can see is that when you go from this original network, you go from about 24,000 connections to, in this shortest path skeleton, you go to about 6,000 connections. In other words, if you assume shortest path routing, you're actually making the assumption that only about, in this case, quarter of the total network density is being used, and the rest is just sort of vestigial. So it's not really a very good use of the resources that you have available. So what I want to take some time then is to consider if communication could occur along other routes as well. So one way that we can kind of broaden our horizons is to think about not just the shortest path between a pair of areas, but combinations of shortest paths and walks, so so-called ensembles. And one very simple way to do that is to consider not just the top one shortest path between a pair of areas, but the top 5, the top 10, the top 100 shortest paths, so the K shortest paths or walks between a source and a target. And really what we're doing here is we're saying, if I pick a pair of areas, I don't want to think about just one communication channel between them. I want to consider multiple communication channels, and I want to ask, what is the kind of the total bandwidth for communication between these two areas? And the advantage of taking this approach is, first of all, I hope it's obvious that it'll right off the bat give you access to other paths and automatically confer resilience, because now communication does not depend on just one dedicated channel. It'll allow more network connections to contribute, so you'll make better use of the resources that you have. And 3, and this is going to be a recurring theme here, that if you start to consider more than just the shortest path, your view of the underlying network, so what's important in this network, the nodes and the connections, could change actually quite drastically. So what's being shown here are the most central connections and the most central nodes when you consider either the top 1 shortest path, the top 50, or the top 100. What you can see is that as you kind of like broaden your perspective and start to consider additional alternative paths, your picture of what the most important connections and what the most important nodes in this network, or the most important hubs, for instance, changes quite a bit. So, I think that's a benefit. And you can take this idea further and really just generalize it to consider all possible paths and walks between a pair of areas. So what is the total communication capacity between two nodes? And this is a really cool statistic that's called communicability. And the way that it works is you have a network, and what we're going to do is we're going to pick 2 nodes, and we're going to find a weighted sum of all possible walks between these 2 nodes. And what we're going to do is we're going to weigh the short walks more and the long walks less. And the way that communicability is set up is for a given pair of nodes ij, you take a sum of the number of walks of length n divided by n factorial. So for instance here, in this particular arrangement, there is no direct connection between nodes I and j. So the numerator there is 0. The walks of length 2, in this case, this is the shortest path. There's only 1, so there's 1 walk of length 2. When we think about walks of length 3, there are actually 2 of them, so that gets added to the sum. And you can do this on and on. So at first it might seem sort of like a very fanciful way of, very sort of idiosyncratic way of weighing these paths, but it's actually quite elegant what Estrada and Hatano did, because, and some of you might recognize this is the Taylor series. So you can very elegantly represent the sum as the exponent of the adjacency matrix, and I apologize I didn't actually say what A was, but A is the adjacency matrix here. So you can write it in 1 line of MATLAB code. So it's quite nice in that sense. So with communicability now, we're really delving more into the realm of diffusion, and now I just want to shift gears and go all the way to the other end of the spectrum and really consider processes that are more akin to diffusion. And kind of the dominant paradigm here is going to be that of the random walk. So with random walks, the idea is we have a network and we have an agent or walker who sits at one node in the network. And this walker will probabilistically move through the graph. So at any given time, the walker will consider who their neighbors are. And they'll have, in this case, in the binary case, an equal chance of moving to any one of the neighbors. So if they have 4 neighbors, the chances of moving to any one of them is 1/4. And then they'll pick one of these neighbors to move to. What you're effectively doing is you're taking the adjacency matrix, which describes the topology of this network, and you're converting it into what's called the transition probability matrix. So what you're saying is, my random walker is currently sitting at this particular source node, and for all of the other nodes in the network, all possible targets, I have a row vector that describes the probability of moving to any one of them. So the sums of all the rows in the transition probability matrix all equal 1. And when the walker finally decides where they're going to go, they move. You then move to that node, and you consider where the walker might move next. In this case, the probability of moving to any one of these targets is 1/5. The walker will choose where to go, move, and the walker will eventually trace out some trajectory on this network. And this trajectory is very meaningful because it tells you then how likely is it for different components of the network, different groups of nodes to communicate with one another. When you adopt this kind of framework, you can actually compute a lot of statistics that are analogous in some way to what we had with shortest path. So you can ask, for instance, how easy is it for nodes I and j to communicate? What is the mean first passage time for the walker to hit node j if it starts out in node I? You can ask, what is the minimum number of walkers that I would need to send out from node I in order to guarantee to within some degree of probability that they actually, at least one of them, makes it to node j, the so-called resource efficiency. So in other words, how strongly would I need to broadcast this signal in order to guarantee that I activate some particular target? You can also ask, if I have 2 walkers that were initiated simultaneously in nodes I and node j, so for instance, the transduction of an auditory stimulus and a visual stimulus, or 2 competing representations, what is the expected time for them to meet somewhere in the network? What is the coalescence time? What is the potential for these 2 sources of information to integrate? And the nice thing about this is that it's actually infinitely extensible. So you can introduce all kinds of cool new additions. You can add biases to the walker, so they no longer just randomly choose where to go to, but they're a little bit more likely to move to certain types of nodes, for instance, based on their receptor densities or gene expression or something like that. You can impart memory on the walkers. You can make sure that they don't double back on their path and where they've been before. And if you have multiple random walkers, you can have interactions. You can set rules for what happens when 2 or more walkers occupy the same node. So now I kind of make this distinction between shortest path routing and diffusion, but we can actually think of shortest path not as a communication mechanism, but as a structural feature. So regardless of how communication takes place, every network has shortest paths. So we can actually ask the question, how easy is it to find shortest paths? And specifically, could a random walker find the shortest path between 2 areas? So if you have a situation like this where you're going from a source to a target, it's trivial, right? Even a random walker could identify this particular shortest path. But what happens in a situation like this where you have to go from source to target, but now the intervening nodes have lots of connections themselves? You're going through a series of highly connected hubs where your random walker could get lost, or in biological terms, where there is a large potential for interference and noise and signal corruption because there's information coming from elsewhere in the network. So the statistic of search information and path transitivity try to address this. In search information, you're asking how hidden is this shortest path. You're asking how much information would I need to impart to the random walker in order to ensure that the walker gets from source to target. And what it really is just a function of the degrees of the nodes that you encounter along the way. With path transitivity, you're counting how many transitive closures there are along this shortest path. So you're asking how many triangles are there, because if there are a lot of triangles along this path, then even if the random walker were to go off this shortest path, they would still have the potential to get back on the shortest path. So the potential for the signal to be preserved or potentially even reinforced is larger. And the reason I wanted to show this is because it shows two things very nicely. First of all, it shows how you can use these kinds of models to make predictions. So what the authors did here was to take structural networks and to compute for every pair of areas the search information and path transitivity. And then they compared this to pairwise resting state functional connectivity in the same participants. What they found was that the greater the search information between a pair of areas, in other words, the greater the potential for the signal to get corrupted as it travels from a source node to a target node, the lower the functional connectivity between them. And conversely, the greater the path transitivity between two areas, the greater the potential for the signal to be preserved as it travels from source to target, the greater the functional connectivity between two areas. So it shows two things. One, how you can make predictions about functional connectivity, but also this distinction between shortest paths as a mechanism of communication, but also as a structural feature that could still be discovered and taken advantage of by other communication mechanisms. So we can actually take this further, and we can ask how well would routing and diffusion work on different types of architectures. So this is actually the same group that tried to address this question. What they did was to grow a set of synthetic networks that optimized either routing efficiency, which is shown on the y-axis. So this is just the characteristic path length, or the diffusion efficiency, which is shown on the x-axis. So this is basically mean first passage time. And they grew these synthetic networks to optimize one of these two criteria. And when you do this, you end up with what's called a morphospace, so a space of all possible network configurations under these particular constraints. And the interesting thing is what happens in the periphery. What happens at the frontiers of this space? So at front one, at the top, what you see is a series of networks that are better at routing than they are at diffusion. And all of these networks, I guess I have a pointer, all of these networks actually have a kind of a specific look to them. They all have these tightly clustered groups of nodes. They have communities or modules. So the reason why diffusion does poorly here is if you have a random walker, a signal, that's being randomly passed from neighbor to neighbor, and all of those neighbors are densely connected with each other, that signal is just going to keep getting passed around in that same clique over and over again. There are going to be very few opportunities for that signal to actually go to the rest of the network. So these kinds of networks are actually very poor at diffusion. But something very interesting happens on front three. Here, again, the networks have a very particular look to them. They all have a connective core. And in these networks, diffusion is actually not disadvantaged. Because in these networks, a random walker could start out anywhere on the periphery. And because they have access to this densely interconnected core, they can easily get to anywhere else in the network just by hitting this particular core. So this is quite profound because it, well, I'll get to that point in a second. But what I want to do is do a quiz. I wasn't able to figure out how to use the app, but I will ask this. So what you'll hear about in the next two talks from Rick Betzel and from Martijn van den Heuvel is that brains actually have both modules and they also have connective cores. So the question is, does routing work better or does diffusion work better on brain networks? So who thinks it's routing? Who thinks it's diffusion? People are still undecided, okay, on the fence, that's fine. So we're in luck because the same authors tried to address that very question. So here, what they're showing are various brain networks and other types of networks. And in terms of their routing and diffusion efficiency, what you can see is that brain networks actually lie very close to the identity line. In other words, by virtue of the fact that they have both a prominent community structure, but also this kind of core periphery organization, they're able to potentially support both types of communication mechanism and to kind of to balance them. So this is really nice because it shows us that, first of all, a communication process and its efficacy strongly depends on the kind of network that it's operating on and what that network possesses, what kind of attributes. And also that potentially there could be multiple communication mechanisms then that are operating in parallel. I just want to close out with one set of models that have to do with spreading. So they're very diffusion-like in a sense. And those are percolation models. So with random walks, we assume that the number of signals, so the number of walkers, is actually conserved. But that doesn't necessarily have to be the case. What could happen is that some signal or perturbation originates in a particular brain area and is then simultaneously transmitted to multiple areas and diffuses along multiple fronts and reaches multiple targets. So one way that you can look at this kind of process is by considering what's called contact percolation. Here we have a perturbation, so a node turns on. This could be the transduction of a sensory stimulus, for instance. And we have a rule. We say that for every node in this network, some proportion of your neighbors have to turn on before you also turn on, before you also adopt the signal. So say that this threshold is 1/3. Basically, what would happen is this particular node would try to influence its neighbors. Each of these neighbors has 3 connections. So from their perspective, 1/3 of their neighbors have turned on. So this is greater than the threshold, and they would turn on themselves. These 2 neighbors would then try to influence all of their neighbors. The 2 nodes at the top, their threshold is exceeded, so they would turn on, but the node at the bottom, their threshold is not exceeded, so they would not turn on, so the cascade would continue and develop. So these kinds of models are really nice because they allow you to ask a lot of interesting questions. They can allow you to ask, for instance, how do signals spread through the network, and how do they reach different parts of the network? What happens if you have multiple signals that are initiated at the same time and that spread synergistically or cooperatively in some sense? Does the cascade accelerate? Or, for instance, what happens if you have multiple signals, but now you endow them with different content? You have both an auditory stimulus and a visual stimulus. What happens, how do they spread through the network? How do they compete for influence? And ultimately, where do they meet, and where does this information get integrated? And you can, like I said, address a variety of questions. You can ask, for instance, which projections are being used at any given time to facilitate the spread of information. You can ask what nodes are the optimal integrators in this network. So for instance, here you can see that the nodes that tend to mediate the intersection of many different signals are all part of the kind of polysensory association or transmodal cortex. So I just want to close out with one final mechanism that doesn't easily fit into the kind of the axis that I showed, but I think is very important to consider, and that is navigation or greedy routing. So with routing and diffusion, they're both fundamental concepts in mathematics, but neither is really completely satisfying as a biological mechanism, right? So when you have shortest path routing, you're making this unrealistic assumption that you know the global network topology. When you have diffusion, you're making this other assumption that signals are sort of just bouncing around randomly in there. So could we envision a different decentralized mechanism, but one that takes advantage of the infrastructure in some biologically realistic way? And so here what you can do is implement what's called greedy routing, where you have a strategy for forwarding signals, but one that does not have access to the topology of the network or knowledge of the topology of the network, but does know something about some different underlying space. So this is a study from Kaius Sagan and Andrew Zaleski and Martijn van den Heuvel, where they said, well, what if you start out with a signal in some source node and you keep forwarding that signal to neighbors that are physically closest in terms of Euclidean distance, they're physically closest to the ultimate target. And what they were able to show is that even with this sort of blind kind of forwarding strategy, and regardless of how you define the network, so for different connection densities, whether it was binarized or not, this particular navigation strategy could actually recapitulate the shortest path structure very well. So that's what's shown with this success ratio variable here. And more importantly, this was much better than a series of both rewired and repositioned null networks. So it shows that that was actually optimal for these for the brain because the brain is a spatially embedded network. And so again, an example of how a communication mechanism could actually take advantage of the underlying infrastructure. So I'm just gonna, I had another slide here, but this is very similar to probably what Martijn will cover. So I'm just gonna skip it and go to the summary straight away. So what I hope I've convinced you is that most network measures assume a very specific communication mechanism. If you open up the brain connectivity toolbox and you start computing measures on your network, you might be agnostic about how the brain exchanges signals, but all of the measures that we use make very important assumptions about how that's done, and it's important to keep that in mind. And when we think about what is the optimal or the most likely communication mechanism in the brain, we have to consider not just the assumptions that mechanism makes, but also the biological plausibility of that mechanism, and also the predictions that it makes about functional connectivity and other properties. Also, shortest paths are clearly a critical substrate or structural feature. Every network has them, but shortest path routing is not the only possible communication mechanism. And indeed, we can envision many other communication mechanisms that can still take advantage of this particular property. And finally, that communication processes on their own, we can consider them on their own. You always have to evaluate the communication process with respect to the network that you're going to run it on, because their efficacy depends on properties of the network, such as whether it has modules, whether it has a core, whether it's spatially embedded or not, so on and so forth. And finally, I think given that the brain does have many of these properties, it's perfectly reasonable to assume that multiple mechanisms could be operating simultaneously, and they could actually be multiplexed in some way. So for instance, different mechanisms are operating in different frequency ranges, or are instantiated depending on where the signal starts, or what type of function you're trying to implement, so on and so forth. And so all of these questions are really just a microcosm of these very complex trade-offs involving both the communication efficiency, but also the cost associated with communication, the amount of information that you would need to impart to the system to get the mechanism to work, the resilience of the mechanism to damage, and so on. So I'll end there and take any questions.
Any questions for Bratislav? So one of the questions that were posted online was in regards to computation of these measures, such as path length on functional versus structural networks. So the specific question is, does it make sense to compute things such as path length on a functional network, given that a functional network, a functional connection in and of itself embodies a communication pathway?
Yeah, I mean, I think you said it right there. I think it depends on what you're trying to achieve. If you're more interested in computing path length-based measures as a kind of getting maybe a biomarker that differentiates patients and controls or makes predictions about some other variable, then it's perfectly reasonable to do. If you're really trying to think about how information is exchanged, I personally think it's much more likely that functional connectivity reflects the exchange of information as opposed to being the conduit for the exchange of information. So it's probably a little bit more, I personally would say it's maybe a little bit more reasonable to compute these measures on structural networks than functional networks, but that's just kind of a personal opinion.
A more recent question that I guess it's a bit more open-ended. The question is, can you please explain links between walkers, travelling waves, and neuronal avalanches? I think it's a very open-ended question, but maybe...
Okay, so...
I guess they really relate to different concepts, right? I mean, a walker is more specific to communication, whereas travelling waves and neuronal avalanches are...
Yeah, I mean, I think that they're probably getting at similar concepts, but at different temporal and spatial scales. I think with traveling waves, we probably tend to think of traveling, maybe I'm wrong, but maybe tend to think of traveling waves as occurring on maybe a continuous substrate. So maybe like a sheet or something like that. With avalanches, I think sort of avalanches are very similar to what I showed with contact percolation. So in that sense, they're also very close to random walk dynamics. It's just that there is really influence is spreading in multiple directions simultaneously, whereas with random walkers, they're sort of the walker is always conserved as it travels, as it hops from node to node.
Thank you. So are there any final questions from the floor? So if not, please join me.
There.
Okay.
Hi, sorry. I was just hoping for a clarification on terminology. Mm-hmm. When you talk about the shortest path,
yes.
The phrase, the source and the target, are you talking about communities of nodes or specific nodes? Is a path between two given nodes, by definition, isn't that the shortest path?
No. So there's a little bit of a difference in how people call this. Sometimes any contiguous set of edges between a source and a target is called a path, and then we think of what is the shortest path between them. I've tried to use this terminology. I actually stole it from Alex and Andrew's book, where they refer to anything that's not the shortest path as a walk, and just the shortest path as the path. But I don't know how communities figure in this, but I will say that one thing that I think something that we don't really study enough is what kind of nodes a signal encounters as it goes from a source to a target. Because it really makes a difference whether you visited just one, say, resting state system in your journey, or whether you visited multiple, whether you encountered nodes that have very similar gene expression profiles along the way, or whether you actually passed through nodes that were very different in terms of their, say, gene expression or receptor densities and so on. So thinking about those kinds of questions might get us to think more about how a signal might ultimately get transformed as it travels from a source to a target.
Okay, great, thank you.
Thanks, Bratislav. So we might need to move on. Please join me in thanking Bratislav once again for the great talk. So our next speaker is Martijn van den Heuvel. Martijn will talk on hubs, rich clubs, and neural navigation. Martijn.
I'm much shorter than Bratislav. So good afternoon, everyone. Great to see you guys all here, and special thanks to Andrew and Alex for pulling the band together and organizing this workshop again. So I think I have the simplest task of all of us this afternoon. How do I progress? My slides do not progress, people from the... Can we... look at that. Good, excellent. So my job is actually quite simple, because we all have a very intuitive way of, very good intuition of what hubs are. So, for example, if we look at transportation networks or flight networks or social networks or computer networks, if we have to think about central elements in these systems and basically their function, what they should do and how they should behave, we all have a very good intuition about this, right? For example, if we look at computer networks, central elements there, they should be important for routing the information between the computers. They are very sensible to damage, right? If one of the central computers goes down, the entire system might be compromised. And in a similar way, we might argue that in social networks, hubs are these people that have many more friends than others, and therefore they act as some sort of a social glue within the system. And in a similar way, we might argue that hubs in biological systems, in particular the brain, might act in a similar way. And as a matter of fact, actually, it's funny enough, we tend to see hubs not just at one scale of brain organization, but we tend to see them almost across all different skills that we tend to study, right? Whether we are interested in gene-gene interactions, or whether we're interested in direct neuronal connections, or whether we are more with, like most of us would do with functional and structural MRI, would look at brain regions and their interactions. So the objectives of my talk would be to say, what are hubs? What types of metrics do we have to some way quantify the existence of hubs? How can we categorize them? What kind of flavors do we have? How can actually hubs be connected them together? And how do they form a central core of a rich club? And I will explain in a little bit more detail what these phenomena are. And I would like to end with a simple notion of how can we compare hubs across your case and control populations. And hopefully, if there's time, I would like to talk a little bit about things that I think many of us in the field are thinking about when we examine hubs. So what are the limitations and perhaps some caveats that these methods have? And what should you look for when you want to apply this in your patient control population? So what are hubs? Well, like I said, we all have a very good intuition about this, right? So I won't spend too much time on this. But intuition is good, but we also need some form of a formal way to define. You say what, this is a hub, and in the end, this is not a hub, right? And I think one of the first notions of this was done by Barabasi and colleagues in their famous science paper in 1999. So at that time, most of the field most of the graph theoretical field was interested in examining the behavior of so-called random networks, right? Like the Erdős-Rényi network, in which nodes are just randomly connected by edges. And if we look at the degree, so the number of edges per node of every one of the nodes in the network, and we make a histogram out of this, this looks a little bit like a bell shape. But what Barabasi noticed is that when he looked at natural networks, like biological networks, like the internet, like transportation networks, he did not observe this type of degree distribution, right? Instead, he actually observed that many biological networks tend to have a 1/f degree distribution. So again, if you count the number of edges per node, then apparently there is not some sort of a mean of a single node would have X connections, but there was a wide bandwidth of some nodes having, actually the majority of nodes having only a few connections, but there were just a few special nodes apparently in those systems that have many, many more connections than others. So, and if you plot that on a log-log scale, right, you see this straight line down. And actually, basically, if we would visualize that a little bit more, we could say, you know what? We tend to say that a network has some sort of a hub organization, or there are hubs in a network, central elements, if there would be a heavy tail. So if we would plot the degree distribution, we relaxed a little bit the constraints of saying, you know what, it has to be a 1/f, but we tend to say that there are hubs if the degree distribution is quite right-tailed, meaning that the majority of the nodes have only a few connections, but there are some nodes in the network that tend to have many more connections, so they're much richer in terms of their connectivity. So then, what metrics do we have to capture this centrality? Well, let's look at this very simple brain network, right? Let's say this is the brain network of Homer Simpson with just a few nodes and a few connections. Then we could argue to say these nodes, I think they're central. And because they're central in a sense that they tend to connect these different modules together, right? So they're the bridges, if you want, between the different modules. But another one could argue that this is a central node, the blue one, because if this node is damaged, then the network actually becomes disconnected. You cannot reach all of the nodes then if the blue node is taken out. So in both, actually, there is some truth, right? Both we could argue that these are the central nodes. So there are actually a wide range of metrics to capture the centrality of a node. And here too, it really depends on the question that you have. So I'm sorry to say, there is no one definition of a node being a hub, or not being a hub. But there are many different types of metrics that capture different properties of centrality. And it depends a little bit on your research question, which type you like most. But some are actually much more easier to understand than others. So our very simple one is degree centrality, right? And we already encountered that before. We just count the number of edges per node, and we do that for every one of the nodes, and then we pick the one, let's say the top 10 or the top 10% of nodes that have the highest degree. A little bit more complex is we would say, no, you know what, we're not only looking at the direct connections of a node, but we're interested in how well a node can send information to another node, to any other node in the network. And we take the average of that metric per node. And this is actually the shortest path length that Bratislav was talking about, right? And we call that closeness centrality. It simply emphasizes how close this node is in the total embedding of the network. Now we can go one level up, right, and say, you know what, we're not only interested in going from our source node I to another node J. We're looking at all possible paths within the network, and then we're counting how many times a certain node is passed in all of these communication paths. And this is the betweenness centrality, right? And betweenness, of course, comes from this node being in between of many communication paths. But we also can have, actually, we can go up one level even further, and when we can look at whether a certain node is central in terms of the overall, the total embedding and the behavior of all of the other nodes in the network. And this is something that is more recently gained a lot of interest in terms of eigenvector centrality or PageRank centrality, which is a flavor of eigenvector centrality. And PageRank, of course, has become quite famous because it's one of the key algorithms of the Google search algorithm. So how does this work? Well, basically, if you take, again, our Tor network, this describes the adjacency matrix, of course, and then if you actually take the eigenvalue and the eigenvector decomposition of that matrix, funny enough, one of the eigenvectors, namely the one that fits to the biggest eigenvalue, is exactly a good metric of how central nodes is in terms of how central the neighbors of that node are. And it actually relates very strongly to the random walker that Bratislav talked about. So again, if we start a certain random walker in a node, and then we let it diffuse over the network, in the beginning, in this case, it has 50-50% chance of picking one of the nodes. And then we do this repetitively over the network, right? So the random walker is walking around the network. And then basically we keep track of how often a certain node is hit by the random walker. So if we keep on doing this continuously, and actually if we do this infinite number of times, you're actually going to end up with a value very close to the eigenvalue centrality, eigenvector centrality metric. And there has actually been some, so you might argue, you say, well, this is quite mathematical, right? So how does this relate back to the underlying biology? And I put strong value to this, right? That I think that the metrics that we study, I always like to go back to the underlying biology and try to find, as far as possible, a link to what this metric could actually truly mean. Well, an example of this eigenvector centrality was actually very nicely done by a study by Boombarger and colleagues in which they compared the neuronal system in the C. elegans worm and the P. pacificus worms. Well, these are two different types of worm species, but they have actually completely different types of behavior. And interestingly, Boombarger and colleagues, using PageRank centrality, could pinpoint this type of differentiating behavior to the PageRank centrality of certain specific nodes in the network. So I think this is a very cool example of how very mathematical properties can actually say something about true biology and behavior. So basically this says we have multiple types of metrics of centrality, right? And I named 4 here, but there are many more, if you will. So we could also say, you know what, I want to define my hubs not in one metric, but and in a similar way I can say, you know what, I think a hub is a hub if this node scores on multiple centrality metrics. Or for that matter, you can also define your own property and say, you know what, I think that a hub should not show a certain property and put that into the mix. And as a side note, it is important to note that if you combine these different metrics, you should realize that they're not independent from each other. Actually, we could put them on some sort of a space, a continuous space. And actually, this one is actually very related to the one that Bratislav showed. And on one end, if we just look at degree, right, we could argue that basically what we're doing is that we're looking at nodes that have a walk of 1. So we're just looking for a certain node, how it's connected to the rest of the node if we only allow very short walks. Now we can go a little bit further and then say, you know what, we allow shortest paths. So we allow paths of shortest length, which would give us our betweenness centrality or our closeness centrality metric. But on the other end of the spectrum, if we only look at some sort of a diffusion type of communication process, and if I'm interested more in that, then perhaps eigenvector centrality or PageRank centrality is much more a metric for you to define the central communication elements in the network. And for that matter, again, like I like to emphasize, there is no one metric that you should use for your hop detection. People have used all sorts of flavors and pinpointed different central brain regions in the connectome. So once we have these hubs, right, then what can we further do with this? Well, one thing that people started to notice is that if we have a selection of our hubs in the network, right? If we pinpoint them by means of our favorite centrality metric, well, let's pick degree for the simplest one. Then actually these hubs might have different roles. You can get a high degree in different ways. So for example, if we superimpose the modular architecture, in this case, Homer's brain apparently has 3 modules, so we plot that on top of the nodes. And Rick will tell us much more about modular detection in the next talk. Then we could actually argue that different types of hubs, so different nodes with a high degree, might have a different role. And this might be something that is quite useful to formalize as well. So for example, if we look at these 2 nodes here, there are 2 flavors of orange, light orange and dark orange. We could argue that the light orange one is much more of a connector hub in the sense that it connects different modules, while the dark orange one is much more of a node that is more central within its own community. And we base this on the type of edges actually this certain node has, right? So in this case, we classify the nodes in terms of the level of different type of edges this node possesses. And like I said, we tend to call the light orange one the connector hub, while the dark orange one, we tend to call them much more provincial hubs. And we can formalize this, of course, right? For example, the within module degree z-score captures the level of connectivity of a certain node within its own module, and basically saying, you know what, if I have a high z-score, that means that I have more connections than I can expect of all of the other nodes that are participating in the same module. And therefore, I am central, in this sense, within my module. Now, the other property of nodes having connections to multiple different types of modules, and therefore perhaps are more integrators of interdomain, intermodular communication, is the participation coefficient. And the participation coefficient basically says, if I have a certain number of edges, then how are these edges distributed evenly or unevenly across the different modules of the network? And a higher participation coefficient in that respect reflects a higher level of intermodular communication of that particular node. So now Germera and colleagues very nicely and very elegantly said, you know what, if I have these 2 different types of metrics on behavior of whether a certain node has much more connections within its own domain or actually across domains, then I can put a space on this and I can pinpoint and classify all of my nodes in the network according to this space. So basically on the x-axis you see here the participation coefficient of a node, and on the y-axis you see the within module degree z-score. Right? So the participation coefficient reflects the intermodular behavior of a node, if you will, while the within module degree z-score actually expresses how well a node is central within its own community. So now if we pick our 2 orange nodes, we can actually put that node into the space. Right? So, and in this case, I highlighted 3 options. We're not going to do this by one of the poll. I'm much more of a human kind of guy, so I like communication with you guys directly rather from a blue screen. So if we pick this dark orange node, right? So where would it fit into the total space? Would it fit in R2, in R5, or R3? Let's vote. Who votes for A, R2? Half. Is that 1? Who votes for R5? Okay, a little bit more. Who votes for R3? 2, all right. And there's no D, people, so let's do this again. You have to humor us a little bit. Let's do it one more time and then pick a node, right? Even if you have not an exact idea, then just try. Give it a try. Who votes for A? Who votes for B? More. Who votes for C? All right. So the correct, if you will, answer should be B, in the sense that this node has a relatively low participation coefficient. Why? Well, actually, all of its connections go to one particular module. So it has, in that sense, no distribution of its edges across the different modules of the network. But if we just look at this within module degree z-score, it should be high because it is much more connected. It's actually the highest connected node of its community. So its z-score, in terms of connectivity, within its own module is relatively high. So let's look at the other one, right? Then where would this one fit? So who votes now for A in R5? Okay, excellent, right? Because it should be different, right? Because, well, it is a completely different type of node. So who then now votes for B in R4? A few more. And who votes for R6? For C? Well, I think I would argue that it indeed fits somewhere between R6 but R4 is probably too high. So why? Well, it is true that this node has a very high participation coefficient. Actually, it has 4 connections, and it connects to all the different modules. So 2 of them go to the light pink module, while the other 2 actually are dispersed across the other 2 modules. So it should have a high participation coefficient. Its edges are almost evenly distributed across the different modules, which would give you the highest participation coefficient possible. Now, it also has a relatively high z-score because the other nodes of the modules are less connected. So then it would fit in R6. And we tend to say that connector hubs would actually be the nodes that are, have a relatively high participation coefficient and also a relatively high within degree z-score. So now let's do an open one, right? When would a node fit in R3 or R4? So when would you have a node that has a relatively high participation coefficient, but a very low within module degree z-score? Does anyone try? Does anyone dare? I see a hand, yes? I'm going to say excellent. I have no idea what you said. I couldn't hear you, but I appreciate your enthusiasm. So excellent job. I would give you an A for this. So actually this node, we would have a very interesting node here. We call them kinless nodes. So these would be nodes that are within a certain module, but mostly connect to other modules. Or it would be a node that only has a few connections and actually are connected to different types of modules. If you have only 2 connections, then it's very easy to distribute your edges across the different modules. So as a side note here, when you compute participation coefficient, and when you do this in the BCT toolbox, and I should here too mention that all of the functions I'm talking about are directly into the BCT, then you get a participation coefficient for every one of the nodes, right? So a high participation coefficient in itself does not automatically mean that it is a high scoring hub. So just think of that when you use this in your research. All right, so as another side note, actually, all of the things that I've been talking about so far were about node centrality, because that was my assignment for today, to talk about hubs. But in a similar way, we can extract these metrics for edges, right? We can look for how central an edge is in communication paths. We can look for how central edge is to certain types of network topological features. And that might be perhaps as interesting to look at node hubs, if you will. So in a similar way, we could argue that there are perhaps some things like edge hubs in the network. Okay, so once we have defined our hubs, then what are the metrics that could capture how these hubs would together behave in the system? So there are 2 commonly used properties that are often described in network neuroscience literature. So on one hand, we have the core, which is the property that a certain subset of nodes should share among them at least k or more connections. So for example, if you look at the highlighted red nodes, they all have, if you would extract this subnetwork out of the total network, then they share among them at least 3 connections. So we call this the 3 core of the network. And this too has been extensively applied in terms of brain networks, both structural and functional, and they tend to show very strong biological plausibility, right? That the most densely connected core elements of the brain would indeed fit to nodes and brain regions that are most often related to multimodal processing. Now we're related, but different. But it is, like I said, related. Property is the rich club. Now the rich club actually is a little bit broader than the core, and it argues that, or it searches for the level of connectivity between the most central nodes in the network. So basically saying, if we have a certain network, and then we've depicted our most central nodes, our most, in this case, most highest degree nodes, then what is the level of connectivity between them? So it could be, of course, below average. Average meaning the overall average density of the network. It could be average in a sense that it is just, what you would expect in terms of their connectivity themselves, or it could be above average, right? In terms that they are much more densely connected than you would expect if you would just randomly pick nodes and edges in the network. And you can argue that if such a structure would exist in a network, if there would be a certain subset of nodes that are super rich in terms of their connectivity globally, then if they also have a super high level of connectivity among them, then they must be really central in the network. Because apparently there's a lot of biological resources spent on keeping these nodes interconnected. And we can very easily capture this by means of the rich club coefficient, which simply counts for any one of subsets of nodes with a certain degree, the number of existing edges, E, divided by the total number of possible edges between those subset of nodes, which in the binary case would be n times n minus 1. And typically we plot this like this, right? So you have on the x-axis your different subset of degree k. Sorry, the x-axis here are different levels of degree. And then you simply count, you compute the rich club coefficient for every one of those subsets, and then you get the, your network of interest, which in our case would be mostly the brain. Now, most typically, we benchmark this to the random network, right? Because here too, actually, if you would have a network with certain high degree nodes in there, then they also already have, by themselves, a relatively high probability of them being connected. And this is shown actually by, if you randomize the brain network by means of degree preserved randomization, then you actually also see a line that goes up, basically, right, over degree. And this is because hubs have a high number of connections, so if I shuffle around my connections, then very likely this edge will end up in another high degree node. So therefore you always have to benchmark this to the random node model, and then typically we take the ratio between the two, right, between our network of interest and the population of random networks. And then you get hopefully a line that is higher than 1, which would indicate that there are a subset of nodes in the network that are more densely connected than you would expect compared to the node model. Now, as a node, the centrality here is based on degree, right? We pick the nodes with the highest level of degree as those that are central, and then we compute the level of connectivity between them. But you're free to use any type of other metric that you have, right? For example, Bertolero and colleagues proposed to use the participation coefficient as the nodes with a high participation coefficient to be perhaps the more central and more important nodes than purely based on degree. And Deco and colleagues have used more computational driven functional models to define nodes that are perhaps important for binding information together, and they call this the binding nodes or the binding club. So I emphasized here again that we should take the random network as the important benchmark here, right? And then here too, there are multiple ways of defining this random network, or null model, as Andrew called it. We could do this degree preserved, or we could do this non-degree preserved. So here too, there are different flavors to do it, and it tailors your exact question that you have in your specific study. So a few final notes on how should we compare nodes and hubs across populations, and then how does this relate back to some limitations that we have. So what do we have to think of when we do case control, mostly hub comparison? So how can we compare hubs? Well, a simple one would just simply say, you know what, I take the degree of every one of the nodes, and then I do a t-test or permutation test between my controls and patients. And I think this is a very simple way and fair way to do it. But people have also proposed more advanced methods. This is a method by Tremenan and colleagues in which they said, yeah, let's take a degree distribution of a healthy population. Let's take ACP data, and we compute the degree distribution. And then I have my new controls and patients, And then I compute for every one of the individual subjects their degree distribution, and I take the correlation between my degree distribution of my single subject to the healthy population. And if it's a good fit, then I find that a marker of hub integrity of the total network, and if I find my correlations to be relatively lower, then the hubs must be disrupted. So, and this was proposed as a hub disruption index. So the deviation of the normal healthy distribution as a metric of whether your hubs are, your hub distribution across your network is healthy or not. All right, so what are the things that we have to think about? Well, the first notion is, so I listed 3 of them. So our first notion is that the metric of degree and metrics of hubness or centrality, they're not independent from the other metrics or other properties of the network. And this was nicely shown by Jonathan Power and colleagues in a very nice, elegant study in which they basically showed that the community size is an important driver for your functional connectivity degree. So if you take your functional network and then you compute functional degree as a metric of centrality, then basically your community property of the network might play a big role in defining which nodes are functional hubs and which nodes are not. And basically the argumentation is the following. So again, we have our toy network here. And then if we look at these 2 different connections, then one is non-existing because we're thresholded it away or it's very low functional connectivity. And these tend to be mostly long range. While the ones with the highest functional connectivity, most of them, the majority, are short range. So now if I have a bigger module, right? If I have a larger module, as you can see in the I have a pointer here? No, yep. This module here, this is a much bigger module. So if most of my connections are actually present within the module, then every one of the nodes that are present within that module will likely automatically have a relatively higher degree. So a node within a larger module, because the nodes are more spatially close, will also have a higher chance of higher FC, and therefore it will end up as having one with a higher degree K in general. While 2 nodes that are in 2 different modules, right, and they're also part of smaller modules themselves, on average the degree will tend to be lower. So when we do a degree selection approach to select our hubs, then most of our hubs will end up in the same module. And the argumentation of Power and colleagues is actually that this might be a little bit of a bias to why we find the FOMO network, for example, so central in the network, because it's simply 25% of the total cortical space. So community size is an important driver for functional degree. Now, a second notion is the influence of false positive and false negatives on this. And I actually was hoping that Andrew would highlight this important paper on this, but I think he didn't, or I was sleeping. But I didn't put much emphasis on this, but then I will do it. I will spend my last 3 minutes on this, because I think this is a very important paper. So basically what Andrew showed in this paper is that having a false positive and having a false negative in network science are 2 different things. And this is very counterintuitive, right? If we do a normal experiment and then we have a false positive and then we have a false negative, and then we say, yeah, okay, that's happening, right? We have false positive and false negatives in any type of experiment that we do. But in the end, I hope that my false positive and false negative will have an equal contribution to the error of my estimation, and therefore having either false positive or false negatives is a little bit of a balance, right? However, this paper shows that having a false positive is almost twice as worse for your network metric as having a false negative. And the very simple reason for this is if you have a false positive, most likely, by definition, it is a random connection in your network, and a random edge in your network will instantly affect your shortest path length, right? It will help your shortest path length a lot. Well, if you just take out one tiny connection in the network, this will not have a strong influence on your shortest path length. So in other words, if you have a lot of false positives, then your network will automatically be a little bit more random, right? Or the path length will be shorter. Now, on a follow-up paper on this, Andrew and I showed that if you actually have a very simple metric of your connectivity density as a whole in the network, in this case, just the total sum of your functional edges in the network, then having a little bit lower functional connectivity, as we normally tend to see in operations, will automatically introduce a little bit more false positives in your mix than than it will do on false negatives. So basically, subjects with a lower functional connectivity will automatically have a few tiny more false positives in the network. But a few tiny more positives in the network will have large consequences for your network metrics. Or put differently, if you have a subject with low functional connectivity, it is more likely to include a few false positives, and as a result, your network will become, you will estimate this network as to be a little bit more random. So now, what is the consequences of this now specific for our hub analysis? Well, basically, random networks, as we showed in the beginning, they tend to have a different degree distribution. Right? If we go all the way back to the seminal work of Barabasi and colleagues, he showed actually that random networks tend to have a little bit more bell-shaped degree distribution. So in other words, if our patients on average have a tiny little bit more lower functional connectivity than our controls, then as a result, we're going to end up with the conclusion that they might have a little bit of a different degree distribution. Or in other words, they tend to have, as we would falsely conclude, a little bit less hubs. And then we would argue that patient and controls would have a different hub distribution. For example, that schizophrenia or autism or Alzheimer or whatever type of disorder you're interested in, these patients might have an affected degree hub distribution. And for example, this will have, of course, consequences if we would simply select our top X% nodes as hubs in the network, and then we could actually argue that patients, for example, have hubs in different places, as a consequence of actually the false positives having a slightly more effect on our network than others. And like I said, this might have influence on the more complex type of metrics that we tend to sometimes want to examine. So in the final thought, let's go back again to the nil condition one more time. So in the Rich Club argument, we tend to keep the degree sequence fixed, right? And the reason for this is that we want to rule out the random chance of hubs being more averagely densely connected to each other, also in the random condition. And the idea here is that we want to keep one property of the network fixed, namely the degree, and then we shuffle the nodes, and then we look for how well our network performs in contrast to this null distribution. Now this might be, and we do this because in the random condition, right, this property also goes up. Now for degree, this is relatively simple, right? We can just shuffle around the edges and use the Maslov-Snapper algorithm. But if we use other types of centrality, between a centrality, participation coefficient, for example, as people proposed, this is quite a difficult thing to do. And to be honest, I have no clue how you can do this properly in a random null model. Because if you randomize the network, even degree preserved, the modules will change, the participation coefficient will also change. So this is not a very simple way to do it. I'm not saying you should not do it, I'm just saying keep track of your null model, because only then you would have a fair comparison and a better estimate of whether your case and controls are really different. So to summarize, hubs are elements in the network that are central, and we can categorize them into different flavors. And as my previous speakers also emphasized, choosing your null model is a very important thing to do in today's network neuroscience analysis. Thank you very much.
Thanks, Martin, for the great talk. Are there any questions for Martin off the floor? I think we have one here in the front. It might be worth, there's a microphone just behind you if you move to the...
Could you please give an example of biological hubs, and if they are involved in pathology, what happens?
So hubs tend to be involved in actually a cross-disorder property, as it now appears to be. So hubs are argued to be biologically expensive. Others have shown that there might have a specific genetic profile with genes that are also enriched for schizophrenia, autism, etc. But there's also been studies that have mapped cross-disorder disconnectivity and then looked for whether hubs are more often involved in all types of pathology, and the simple answer is yes. There was a very nice meta-analysis paper by Crossley and colleagues a couple of years ago which very nicely showed that. And the idea here is, I think, very intuitively, again, linked to what a hub is, right? A central element. So if there would be damage in hubs, then it would have disproportional effect on the overall system.
Any other questions off the floor? So perhaps we'll take one of the online questions. And the question, well, it's a bit of a long-winded question, but in a nutshell, it's saying if we put aside all of the typical things that are associated with reliability, such as the quality of the data and head motion and so on, is there any specific parameters that are associated with reliability of determining centrality in hubs? So what dictates whether hubs can be reliably found across data or across subjects?
I'm not sure whether there are a really specific type of, I think all of the metrics that we examine tend to be so closely related to each other, and most of them are based on degree, of course, in this case, or strength. Well, perhaps one of them could be that actually, so in my book, I think hubs are a very exclusive set of the network. So if you just make a degree distribution, there is always a top 10%, right? And you could actually say, you know what, I'm taking the top 30%. So I take almost a third of my network and call them hubs. And you could actually call 90% hubs, of course, right? But I think, fitted to our intuition, hubs should be a relatively exclusive set in the network. And I think not many other metrics are sensitive to being an exclusive set of the network.
Okay, any other questions for Martin? If not, please join me in thanking Martin once again for the great talk. So our next speaker is Rick Betzel. Rick's from Indiana University, and he'll be speaking on modules in structural and functional brain networks. So without further ado, Rick.
Yeah, thank you for having me. And as Andrew said, I'll be talking about modularity in brain networks and mesoscale structure. So you've heard a lot today about network metrics, right? And we tend to dichotomize network metrics as being part of different scales. You know, on one hand, there are node level measurements. They can be as simple as properties of a node, like the number of connections it makes or its centrality. And on the opposite end of the spectrum are networks that say something about the network as a whole. The ability for information to travel efficiently from one part of the network to another. But situated between these two extremes, these local and global scales, is a rich mesoscale. And at the mesoscale, we're not focusing on properties of individual nodes, we're not focusing on properties of the network as a whole, but we're focusing on subnetworks. And we call these subnetworks modules or communities. The division of a network into a series of modules or communities we refer to as a partition. Why do we like mesoscale structure? Why do we like a network to have modules, to have communities? The first simple reason is it's a form of dimension reduction, right? Rather than having to describe every node in the network, which is a big, long list, a list of every node and every edge, we can describe the network as a series of communities, much lower dimensionality. We also like mesoscale structure because we think it tells us something about network function. Groups of nodes with lots of shared connections, they ought to be functionally related to one another. And this is borne out in a lot of analysis of real brain data too. And as Martijn already said, we can use mesoscale structure as a way of identifying influential, important, or central nodes in the network, for example, by hub classification. It's a technique, studying mesoscale structures. It's a technique that we can apply equally well to functional and structural data. And importantly, from an applied perspective, mesoscale structure has been shown to covary with disease state, cognitive state, fluctuates over time. So there's lots of applied reasons why we should be interested in mesoscale structure in networks. This is where the challenge arises, right? We want to study mesoscale structure, but given a new brain, given a new scan, given a new individual, how do we actually determine what that mesoscale structure is? How do we figure out whether our network has communities or not? And in general, this is a tough problem, right? Because just by looking at the network, especially if it's a really, really big network, we can't determine whether it has modules at all, right? So here's an example of a network, right? It's an adjacency matrix. Every black dot represents a connection. Looking at it, it looks like scrambled noise, kind of. There's not a lot of structure to it. But if we were to reorder the rows and columns, magically, these modules, the structure emerges. This mesoscale structure emerges, right? So in practice, we can't determine whether a network has modules based on visual inspection. We need to use algorithms. We need to use quantitative tools to do this. And there's a lot that goes into these tools. The user has to choose an algorithm to determine what constitutes a community. The problem tends to be computationally intractable. And almost all of the approaches that we use, all of these algorithms, all of these methods, they're sensitive to parametric variation. Some of these parameters were forced input just to use the tool. Other times, the parameters are kind of buried in code that you may not even be aware of. But it's a tough problem. So for the rest of this talk, I will describe one particular tool or one particular framework for detecting modules in brain networks. And it's related to maximizing a modularity heuristic or a modularity function. And I'll walk through two examples. The first where we apply modularity maximization to structural connectivity data. I'll talk about how modularity maximization can be extended to multi-layer networks. And I'll follow up with an example where we apply it to some time-varying functional connectivity. I know Sarah is probably going to talk a lot about this, so I'm going to be bare bones on that. I'm going to be really quick in that section. And then if we have time, I'll describe some additional reading and some caveats. I'll also add that I'm going to posit a couple points because I'd like to have an opportunity for questions to be asked. Like Bratislav, I couldn't figure out how to embed those questions in here. But in roughly halfway and then a little bit later, if you have questions, shout them out and we can talk. You'll see when it comes up in the slides. And so the goals of this talk is not to give you, a definitive roadmap for how to do community detection. It's a hard problem after all. Rather, my goal is to convince you that community detection is hard. It involves lots of decisions that a user has to make, and these occur at different steps in the process. And as difficult as these decisions might be, as numerable as they might be, the fact that we have some hand in choosing communities and choosing these parameters, it actually makes the technique more flexible and accommodating to different classes of research questions. So I'll start with a description of the modularity function, and then I'll build into the first example. So modularity maximization is one technique for detecting communities, but in reality, there are many. And some of these you may be familiar with. InfoMap, for instance. If you ascribe to the wash you mentality, this is your tool for detecting communities. And that permeates to a lot of the neuroimaging field. So InfoMap is a way of detecting communities based on the flow of random walkers moving over a network. On the other hand, there are other techniques that allow for overlapping communities like clique percolation, or linked communities that actually don't classify nodes in terms of communities, but actually classify edges. And there's even more exotic inferential techniques like block models that have been explored in the context of neuroimaging data. But rising above all of those techniques is modularity maximization. So this is the original paper. This is from actually last summer. And you can see that as of 2018, it had been cited over 10,000 times, so it's awfully popular, right? And this is not just neuroimagers who are using it, but it's a widespread technique. So how does modularity maximization work? It's actually quite simple, and it defines communities to be groups of nodes whose internal density of connections is maximally greater than what we'd expect by chance, or chance is some kind of null model. Right, so what are the ingredients for modularity maximization? First, we need our observed network. So this is an example from structural connectivity derived from diffusion imaging. I'm going to label this matrix as A. We need some intuition of chance. We need some kind of random process for generating a network, some null connectivity model. And we'll talk more about what these models look like. But in general, it's some model that when you run it, it produces a synthetic network that has some desired set of properties. If we were to run it an infinite number of times, it would produce the expected weight of every connection, right? So this is for every element in this matrix, it denotes the expected weight of that connection under this null connectivity model. The modularity maximization works by simply subtracting these two from one another, observed minus expected, and it gives us this matrix B, which we'll refer to as the modularity matrix. And our goal is to choose clusters so that those clusters are internally dense in terms of the elements of this modularity matrix. So on the left, you see an example of a low-quality partition. So these blocks represent communities, and you can see that in terms of the internal connections, there's some red elements within each community, but there's also a lot of blue, right? And so that means there's a mix of stronger than expected and weaker than expected. This is an example of a low-quality partition. On the right, on the other hand, is a high-quality partition, where within the communities, almost every connection is really bright and really red, indicating that it's much stronger than we'd expect by chance. So this is the kind of intuition that we're striving for. We want to get rid of low-quality partitions. We want to seek communities that correspond to high-quality partitions. This intuition can be formalized really nicely through the modularity heuristic. So Q, in this case, this is the modularity function. This is some function that we want to optimize with our community detection. It's the quality. It indicates how good our communities are. Also in this formula is AIJ, which tells us the observed weight of the connection between two nodes I and J. PIJ tells us the expected weight of that connection. And then these variables GI and GJ, it indicates the community to which nodes I and nodes J have been assigned. So our goal, essentially, is to choose these communities such that Q is as big as possible. And then also this delta function, it's equal to 1 when those two communities are the same. So effectively, this summation is over all node pairs, but this delta function means that we're only adding pairs of nodes to Q if they fall in the same community. And again, the final value of Q, it kind of tells us how good a partition is. It's the quality function. So our goal is to choose those community assignments then so that Q is as big as possible. And how do we actually do this in practice? Well, it turns out there's lots of different ways. Lots of algorithms, lots of heuristics, some based on agglomerative clustering, some based on spectral decompositions, some based on simulated annealing. But by far the most popular is the so-called Louvain algorithm. It's both fast, it's accurate, and it's a multi-step greedy algorithm. And I'll walk through it briefly. It's initialized with every node belonging to its own community. Gradually, those nodes are merged into communities as long as it increases Q. When no mergers can increase Q further, those communities are aggregated into meta nodes, and the process repeats itself. And so you can implement this in the brain connectivity toolbox using the community Louvain function, or a personal favorite, the gen Louvain function, which you can download here. Okay, so let's look, how do we actually do this? Let's go through an application. So we have some network. This is the same network from before. I've changed the color map so it's not red, it's a little more appealing. We have our quality function. We have our optimization method. So what do we do next? This should be an easy problem, right? Right, so AIJ, that's our matrix. This is what we want to optimize. We want to choose those community assignments. But the first question we're confronted with is, how do we choose our value of PIJ? How do we determine what our correct null model should be? And this decision is non-trivial. It's not something that you can skip, really. It has important implications for the character of the detected communities, right? This is ultimately because the null model is determining which of those communities, which of those connections are considered expected or unexpected, right? And like so many other answers to questions today, the correct null model, it really depends on your research question. It really depends on the context in which you're using modularity maximization. So on one hand, you could choose a flat null model, where the probability of forming a connection is flat over the whole brain. This would correspond to an Erdos-Renyi random graph, which has been talked about multiple times today. You could also build in a null model in which node's degree or strength is preserved. This is the configuration model, or the so-called Newman-Gervan model. And this is probably the most common. I think most applications end up using this degree-preserving model, although I should add that this particular model is totally inappropriate for correlation matrices. As Andrew mentioned earlier, correlations have transitive properties, and they assume the independence of every connection. This degree-preserving model, this degree-preserving model assumes that every connection is made independently, whereas correlation matrices do not. So this is an inappropriate model for most correlation matrices, for correlation matrices. But of course, we could also build in more complicated models that say preserve spatial relationships, right? The correct model for you depends on context. It's a personal decision. It depends on how you're using modularity maximization. All right, so let's say we've decided, just for the sake of application, that we'll use this degree-preserving Newman-Gervan null model, right? So we run our Louvain algorithm. The first time through, it gives us a quality of 0.4. It's some number. It tells us how good the partition is. Here's our network, and here are the community assignments. We can see that there are 4. There's a red, there's a blue, a cyan, and yellow. And these indicate the communities to which each node is assigned. When we reorder the matrix according to those communities, it looks pretty reasonable, right? Each community tends to be internally dense. The connections are strong within each community. So it satisfies our intuition, right? This is what a community should look like. They're dense subnetworks. But let's just say for kicks, we ran the algorithm again. And we get another equality of about 0.4, Q is about 0.4. And we have a new set of communities. And importantly, these new communities, they're not the same as what we had before, right? So almost identical qualities. They're just as good as the previous set of partitions, or the previous set of communities, but they're different. And when we reorder the network by these new communities, again, we see that they're different. They're giving us different sets of communities. And we can see this again when we compute the community allegiance matrix. Some people call it an agreement matrix, a co-assignment matrix. But it's basically telling us how often every pair of nodes is co-assigned to the same community. If these two were identical, these two partitions produced identical communities, every element in this matrix would be either a 0 or a 1, but in reality, there's some nodes that are split between the two assignments. So there's some intermediate colors. So they're not identical. Why is this? What is the explanation? It has to do with what's called the near degeneracy of the modularity landscape. And it turns out that there are, in general, many near optimal, but potentially different partitions. And it has to do with the fact that if I had a good partition, imagine you just switch the community assignment of one node. You haven't changed the partition that much. You probably haven't changed the quality of the partition that much, but it is a different partition nonetheless. And so this is looking at a set of partitions embedded in two-dimensional space, and at the top, you can kind of see this plateau of all really, really good partitions measured by Q, but they're also dissimilar. They're not situated at the same point in two-dimensional space, right? So this is a problem a little bit, right? We run the algorithm multiple times. We have many good but dissimilar partitions. How do we go about resolving this? And actually, if I were to rerun this 1,000 times, right, we have many different partitions, most of them not identical. And here's the community allegiance for that, for these 1,000 partitions. So they're not identical. How do we resolve this variability? The standard approach is to actually take the allegiance matrix, or the co-assignment matrix, and to start clustering that. So we run the algorithm, we run the Louvain algorithm thousands or hundreds of times, obtain a co-assignment matrix or allegiance matrix, and then cluster that. And this is a procedure known as consensus clustering. And the communities that we get back are groups of nodes that have strong community allegiance. So if we have our observed allegiance, we permute the module assignments give us an expected allegiance, and we cluster this many, many, many times, and it turns out this will tend to converge to a set of consensus partitions. So this is a strategy for resolving variability in the output of your community detection algorithm, right? And so the consensus is perfect in this case. All right, so now that we've done this, we have our consensus partitions, we've resolved that variability. Surely we're finished. This is all that goes into detecting communities, right? Turns out that's not the case. Modularity maximization and InfoMap 2, it has what we call a resolution limit. And what the resolution limit tells us, it tells us that even for networks that have unambiguously defined communities, if those communities are small enough, they may not be detectable by the algorithm. This is kind of counterintuitive, right? So essentially what I'm saying is imagine we had a network like this, of a bunch of cliques, so these are fully connected subgraphs. Intuitively, our expectation is that if I were to run community detection on this network, I would get back something that has like this character of communities, right, where each clique is its own community. But in reality, we get back something that looks like this, right? So modularity maximization is failing to return what we expect. It's giving us something that's a little bit off. And the reason is because if we have maximally defined cliques and a sparse enough network, the fact that there's even one connection between them, it's much more, it's very unexpected. So from the perspective of maximizing Q, it makes sense to put adjacent cliques into the same community because that binding connection is so unexpectedly strong. But the caveat here is that the modules that we detect by maximizing Q, they may not reflect the intrinsic or true mesoscale structure of a network. So we need some way of addressing this. Now, one possibility is to add what's called a resolution parameter to our modularity expression. And what this does is we add this gamma value here. So this is the original modularity, and on the right is the multi-scale or multi-resolution modularity. So all we've done is add a gamma, and this gamma is added multiplicatively, so that's scaling the expected weight of a connection. And if we vary this value systematically, these are the modularity matrices we get. So this is when that gamma value is 0.2, so the null model doesn't count for very much. We make it a little bit bigger, the null model counts for a little bit more. We make it even bigger, the null model is more important, even bigger. But effectively what this does is it changes the number of communities that the algorithm detects effectively, right? So as we change gamma, the number of modules, the number of communities increases, while the size of the module, in terms of the number of nodes, decreases. So this is a way to effectively overcome the resolution limit. And there's lots of strategies for choosing the correct value of gamma. It can be done heuristically, it can be done by cross-validating with an additional data set. Or if you're like myself, you can embrace the multi-scale approach and say there is not one particularly privileged value of gamma, but rather we should consider an ensemble of different values. But the important thing here is there is a parameter that allows us to scale the importance of the null model, which allows us to detect communities of different size and composition. And I should add that there is some lingering idea that gamma of 1 is a special value or privileged in some way. It's the default value that's sometimes buried into other people's modularity maximization code. It is not special, in fact. So by choosing the default value, you're not addressing this question. You're just punting on it and agreeing not to discuss it. So that's an important point to make. So a quick summary of this first part. There are many methods for studying mesoscale structure. The modularity function, Q, is a quality function that tells us how good a partition is. Large values of Q correspond to internally dense and externally sparse communities. Maximizing Q is, in general, computationally challenging, so we're forced to use heuristics, like the Lavon algorithm. In order to do this, there's some requirements that we need to address. We need to come up with a good null connectivity model. We need to, in some way, deal with the fact that the algorithm, like the Lavon algorithm, is non-deterministic, and that there exists a degeneracy or a near degeneracy of optimal solutions. And we also need to consider the scale or the resolution at which our modules are being detected. So at this point I'll pause. This is the first pause. So if there's any questions, I'd be happy to try and address them now. Or if there's no questions, I can move. Yeah, there is a question. Of the what? Yeah, yes. I know I abuse this occasionally, and other people do too. The quality function is only relative to a particular network, is only relative to the network that you are studying. Like the absolute value doesn't mean a whole lot. So the fact that it's decreasing, it necessarily has to because there's fewer and fewer connections above the expected weight. So it's just a way of, you can look at it as just a way of tuning the scale. It's just a way of tuning the number of communities. Q has to necessarily decrease because you're making fewer and fewer connections supra-expected. This is a complicated question. I'd be happy to talk afterwards, actually. I know where this goes. I've had this conversation a lot. And yeah. Yes.
Yeah, I have two questions. The first is when we look at the brain and look at the modularities, is it always important that the network is connected, or is it also okay when we have separated modules?
Yeah, so in the case of modularity maximization, it doesn't have to be fully connected. It does not have to be.
Okay, so I can threshold my adjacency matrix and binarize it, and then I have a separated cluster, and that's also fine then.
You absolutely could, although I would argue it ceases to make sense in some ways. Like if you wanted smaller or larger clusters, you could also just keep a fully weighted network and just tune the gamma parameter too.
Okay,
Yeah.
and for the gamma,
Yes.
how many modules would you expect in the brain network? Because with the gamma I can choose the
Yes.
size. So how many would the brain be?
Another very difficult but important question, and I think it comes down to, I think we think of brains as multi-scale. I don't know that there is a predefined number that I would expect. On the other hand, the whole point, or at least one of the points of doing community detection is to get some lower dimensional representation of your network, to discover some of those lower dimensional structure that's present in your network. So if you have n nodes and somehow you have n modules, that's not very useful. It's similarly not useful if you only get one module back. So the answer is I don't think there is a privileged number, but it's certainly fewer than n and more than one. Maybe a good rule of thumb is like if you're like, I don't know, what people report in the literature is like usually no more than 20 or 25 or so for most parcellations. I think that's the most that I see.
Is there any way how to when a reviewer asks you why did you choose 20 modules, what would you say?
You could try some kind of cross-validation. You could approach the fact that there may be multi-scale or hierarchical structure in your network, so that instead of focusing on a single gamma or a single resolution, you report values for a range that are by some other criteria deemed to be important at different,
Okay.
maybe, yeah.
Thanks.
Yeah. Oh, yeah. For what kind of detection? So the question was, has modularity maximization been used for anomaly detection? Anomalies in what? Can you be more specific? So I'm sure that there's been a comparison of community structure for epilepsy patients and controls. I don't know what the outcome is because that's not my area, but I'm sure that there's been an application. Yeah.
Another question regarding the number of nodes. Is there an interaction from your experience based on the data you use for matrix generation and then from communities you can detect or reliably detect as well over scales? Because I would expect bad data gives more noise in different scales.
Yeah, so the question is just based on the nature of the data, is there some expectation for the number of communities? I think a good approach to for addressing that question is to actually look what's in the literature. So there's a lot of people who are arriving at the same solution with very, very different acquisitions, very, very different processing streams, very, very different analysis pipelines. And that's why I said something below 20. It's like the canonical resting state networks if it's fMRI. If you're detecting those, I would say you're probably doing okay. Community detection is a special form of clustering, and clustering oftentimes is more art than science at times. Like it's a lot of heuristics that are kind of one-offs. So I resist saying anything definitive. In fact, that was one of the points of my talk was that there is no optimal pipeline for doing this, or no optimal number, no optimal. It's not a plug and play kind of approach, but that there may be there's room for some user experimentation, I think. Yeah. I'm going to pause. We can talk. I'm actually going to skip the multi-layer stuff because I know Sarah is going to talk about it, and I'm looking at the time, and I have 6 minutes left. So I'm going to skip the multi-layer. I promise you Sarah is going to do a good job because I saw her messing with her slides when I was sitting next to her. So they look very comprehensive, and there would be a lot of overlap in terms of what I'm going to say and what she's going to say. So I'm going to skip these because they're very, very similar. Okay. But I do have a caveats slide, and I like to talk about that before we end. So this is the additional reading. This is some additional thoughts that should guide your exploration of community structure in structural and functional brain networks. Yeah, so things I didn't talk about today, besides the multi-layer, was how to compare communities across individuals. I didn't mention that totally random networks with no community structure can have very large Q values. So you can detect big values of Q when there is no true structure. This is a problem, of course. This is why I was suggesting that Q itself may not be a good metric for determining whether your network is or is not modular. I also didn't get into any details about how to choose the resolution parameter gamma. But I want to talk about two other caveats really quickly. And the first is it's true of modularity maximization, it's true of InfoMap, and it's true of most community detection algorithms, especially as they're being used in network neuroscience and neuroimaging. When we're trying to maximize Q, or we're trying to reduce the map equation in InfoMap, those tools, InfoMap, modularity maximization, they are assuming that your network has internally dense and externally sparse communities. It is seeking out that type of structure. It's making this assumption. If your data does not have internally dense, externally sparse communities, then you are shoehorning your data into a model that may not be appropriate. Right? If your network is not composed of internally dense, externally sparse subnetworks, you're pushing data into a model that doesn't really describe your data. And in fact, we can see lots of different kinds of mesoscale structure in networks that are not internally dense and externally sparse. For example, core periphery is a class of community structure. It's a class of mesoscale structure where there's an internally dense core that projects to a periphery, projects back to the core, but the periphery doesn't talk to itself. If your network has this and you push that into modularity maximization, I don't know what you'll get. It'll give you answers, but it won't describe the true structure of your network. Similarly, you could have something totally different than the internally dense and externally sparse. The exact opposite, like a multipartite or disassortative structure, where the connections are preferentially falling between modules, so there is no internal connectivity, right? Or you could have some mixture of the internally dense, core periphery, disassortative. You could also imagine more complicated motifs. But modularity maximization and InfoMap are not looking for this type of structure. They're not looking for core periphery or disassortative or this mixture. They're making a very strong assumption about the nature of the communities that are present in your data. This is worth keeping in mind. We now have a couple papers exploring alternative techniques that are capable of detecting different classes of structure simultaneously. Other people I know are using this. So it's worth thinking about these alternative methods as well. And there's some code to do this on my lab's website too. And then finally, I want to note that community detection, this whole process, this whole endeavor of trying to cluster networks, it's actually an ill-posed problem. It's a problem that has no unique solution, which is kind of, it undercuts the point of my talk in some ways, because ultimately a lot of us are users, we want to get the communities in our network, but it turns out there is no uniquely optimal solution. And I'll explain why briefly. So imagine we have this big capital G. This is our graph. This is our brain network. It's composed of edges. It's composed of vertices. There is some function that generated that network. There is some function that distributed those connections the way they are. That's the little f, right? It's some function, it's some generative function of our network. What community detection is trying to do is invert that process. It's trying to say, I have my network. What was that function, and what were those communities that gave rise to my network, right? This is essentially what community detection is trying to do. But it turns out that if we want to invert this process, there may be, and in fact there are, we can always come up basically with an infinite number of functions that map our observed network back to different sets of communities. There is no unique function that does that, right? Essentially, that's the point. And this is proved quite nicely by Ledo Peel and Aaron Clauset recently. So essentially, unless we know the process that generated our network, unless we know that definitively, it is impossible to say with 100% certainty whether the communities we detect are the correct communities or not. And I realize, again, that kind of undercuts my talk, but it's an important point to consider, and it motivates exploring other techniques, like the ones I was mentioning before, that may seek out non-segregated communities. So with that, I want to end and say, just repeat what I was trying to convince you of today. I wasn't trying to give you a roadmap for detecting communities and modules, at least not a definitive one. I wanted to convince you that there were some important steps that you need to think about along the way, namely that this process is a difficult one. You need to choose null models, you need to choose parameters, you need to choose the actual method itself. And as challenging or as dissatisfying as this may be, the fact that we get to make these choices, the fact that there are choices to make, it actually makes detecting modules more fun and capable of addressing different research questions that may be of interest to you. So with that, I will pause, and if there's any lingering questions, I'm happy to take those now.
Are there any questions for Rick off the floor? Yep, at the front. Do you want to just go across to the microphone? There's a microphone behind you.
Thank you for a great talk.
Thank you.
I'm clinical neuroscientist, and I often encounter a situation I have to compare between two samples, and then whenever I used this modular detection in patient and controls, I always wanted to compare how these modular structures, the detail of these structures are different between this. And then I kind of know that there is no also clear solution for that, but if you have a better opinion for this, please.
Yeah, so modesty should forbid me from saying this, but, and maybe Sarah will touch on this in her talk again, but we recently had a multi-layer extension of community detection where you treat each layer, each subject as a single layer, and by optimizing the community structure for all of the subjects simultaneously, it actually automatically maps community similarity from one subject to another. So it'll automatically tell you, at least from the perspective of the algorithm, whether community in one subject is the same as in another subject. I'd be happy to talk about this offline. We have a bioRxiv preprint where you can look at how to do this, but it's difficult to map communities from one individual to another or from one condition to another. And there's ambiguities that can arise in that actually makes it really, really difficult. Our approach is one approach that may help solve that, but I'm sure there are others that I'm not aware of. Yeah.
So I might wrap up with one of the online questions. So the question is, how does modularity sit within other clustering techniques? I think it's in terms of, for example, agglomerative clustering, k-means, and so on.
Yeah.
So where does modularity sit within?
Yeah, so modularity maximization is a tool that is very similar to the other techniques in that it's seeking a hard partition of your network or of your data. So every element, in this case nodes, are assigned to one cluster and one cluster only, just like, say, k-means or other distance-based clustering approaches. But unlike those approaches, this is tailored specifically for networks. In this, it's not computing a similarity-based cluster. The clusters aren't derived on the basis of similarity, but rather on properties of the network. So I think that's probably the biggest difference between modularity maximization and k-means, for instance. K-means has also been studied longer, so you'll probably find lots more variations and lots more deeper dives into something like k-means than modularity maximization. But I think the biggest distinction is that one is suitable for graphs, the other is, you could apply it to graphs, but it wasn't designed that way.
All right, so thank you so much for having me and organizing this. I'm going to spend today talking about multi-layer networks and how we can use them in neuroscience data. So as everyone else here, basically, I was not able to figure out the interactive system, so I'm going to take a little survey by hand. So how many people here have heard of a multi-layer network? Okay, good. How many people have actually used them if they're doing a network analysis? All right, not nearly as many. Okay, good. So I'm going to spend a lot of time kind of motivating why we would use these and going through the simple steps of how we actually build networks. I tend to think of this really as more of a modeling problem, so you're going to get a lot of my personal opinions on building models and how to do that, but hopefully these are things that you can take into your own research and think about when you're designing how to build multi-layer networks, why you're making certain choices. All right, so the reason we're interested in multi-layer networks generally in neuroscience is that I don't think anyone here actually thinks of the brain as a static network, right? We're always doing something. Cognitive processes are dynamic, all right? And maybe we're capturing brain activity over some period of time, but even if we think about structural networks, where we've talked about these tractography networks, those evolve over time, okay? It's a longer time period maybe that we need to look at, but the brain is constantly changing. All right, so there's multiple ways that we can think about the brain as changing, and multi-layer networks are really going to give us a very, very flexible framework for thinking about how to think about the brain across a whole variety of dimensions, be it time, be it different subjects, be it something else. This is a framework that's very flexible, okay? So in my talk, I'm going to go through kind of two basic, I guess, the first half and the second half. So in the first half, we're going to focus mainly on what is a multi-layer network and how would we build them from neuroimaging data. Okay, so during that part of the talk, please be thinking of questions because then we're going to pause and I'll answer questions about building networks. And then we'll talk more about, well, once we have our network, how do we think about measuring things that might be meaningful? Okay? All right, so first we'll start with what is a multi-layer network. And I'm going to start by taking a step back from brains because I think it's a little bit easier, if you haven't heard of what a multi-layer network is, to think of it from a more generic standpoint first, because I really want you to see how flexible this model is, okay? So when we think of the normal networks we've been thinking at, that would be a single layer in one of our multi-layer networks. So this picture here is an example of a multi-layer network. So one of these bluish layers here would be a single network. So nodes in this network are the same. They're these circles. And these dark solid lines represent what we would think of as normal edges in one of our static networks. Okay? However, in the multi-layer framework, we don't have just a single network. Okay? We have multiple networks. So we have nodes, we have layers, and then we have aspects. Okay? And an aspect is something about the feature of an edge. So it's telling us what type of interaction we're defining. All right, so today we've heard a lot about structural networks or functional networks. Those are two different ways of defining edges. All right, so those are two different aspects that we could think about. Or you can think about this as different dimensions of how we're going to build these multi-layer networks. That said, in this slide, I think it's still a little confusing. So I'm going to go to a very specific example. And this is from one of the review papers that first came out trying to define multi-layer networks. So this is an example about network scientists and the conferences they were at and the types of interactions they had at this conference. And this just gives you an idea of the different ways you can combine layers. All right, so each one of these nodes represents a person, okay? And then if we go up, the green, blue, orange here, these are different conferences that people attended. And the this, I guess, other axis here, there's an edge if two people talk to each other, all right? And in this dimension over here, there's an edge if the two people went to a talk that the other one gave, okay? So they're defining two different types of interactions. And then you can see there are all of these dotted lines, okay? So the solid lines are going to be our intra-layer edges, and these dotted lines are inter-layer edges, okay? So they're linking nodes between networks. So in this case, what you see here is these are self-identity links, all right? So there's an edge between, this is AC is Aaron Closet, all right? So there's an edge between him and all of these layers to identify that that node is itself, okay? The other thing I want to point out is that not all of these layers have the same nodes in them. Okay, so there was a question, I think, in one of the first sessions this morning about had people looked at methods where maybe over time you would define the nodes differently. And this is a framework that allows you to do that. Okay, there is no restriction that all of your nodes be present in every layer. Okay, you're going to define edges between them to kind of tell you who's where and what's going on. All right. Okay, so this is an example not of the brain, obviously. So how do we think about this when we think about brain networks? So now this is being drawn in a completely different way, because there's many ways to visualize this. But in this case, we have an axis of brain regions, okay? And then we have time in our other dimension. Okay, so these are interactions between brain regions at time point 1, at time point 2, at time point 3. So now we're getting the idea that this within layer would be a slice here. So you can see in this middle region here, this slice of time 4, that would be one of the layers of our network, okay? Or we could even think about taking a slice the other way, okay, just on a different dimension. And we could ask, well, what about brain region 2? How does that evolve over time? Okay, so kind of how we slice this multidimensional network is going to let us look at different features of our network and how they evolve across these different dimensions. So here you can make it even more complicated, right? I can take that single network I had that was brain region as a function of time, and I can say, well, now I'm examining many subjects over time, right? So I'm adding another dimension where I'm going to be linking not only brain regions and time, but I'm also going to be doing that across a bunch of different people. Okay, so in this case, I might have these dotted lines here that are again saying this brain region 5, is brain region 5 in this network and in this network and in this network, okay? So you're going to have this idea of layers, and you're going to have these interlayer links going between them. So that's a really flexible framework, all right? I've described many different dimensions that you can look at, but it also makes it really complicated to think about how we would actually do that type of modeling and measure things on the network. Okay, and so one of the great things about multi-layer networks is it is so flexible and you can do so many things with it, but also one of the realities is because it is so flexible, a lot of the math hasn't been figured out for how to do that. Okay, so I'm going to take a step back when we talk about this for brain networks, and I'm going to talk about just sequential graphs today. So I want you to understand that it's this very flexible framework, and every week people are coming out with more ways to think about, well, if you add some sort of complication to this, how could we design measures? But some of the most studied measures, and where you're going to find a lot of the math has actually been worked out, is looking at these sequential graphs, where we're just going to take kind of a single normal static network that we would think about and add one dimension to it, okay? So in these pictures, you can think about, again, a sequential sequence of graphs here. And then we can define, specifically for brain networks, we might be interested in something like temporal networks or task networks, okay? And there's a difference between these two types of networks. When I say temporal network, we're assuming that it matters how I order these graphs, okay? We have time point 1, we have time point 2, we have time point 3, we have time point 4, and they go in that order, okay? So the way that we're going to represent having order in this graph is we're going to take each node and we're only going to link it to itself in the next layer. Okay, so that's going to give us this idea that there's some flow of time throughout our layers, because I can't take this node here in the top corner and I can't jump to time point 3. I have to go through time point 2 to get to time point 3. However, if I have some sort of data that's maybe different tasks in my fMRI data or something like that, does it matter the order that the tasks happened in? Right? Maybe in some cases it does, but in most cases we'd want to think of that as something where time actually doesn't matter. Okay, so in that case what we'd want to do is we'd actually want to link a node to itself in every layer. Okay? So you can see that here, where each node links to itself in every single layer. All right, and this is something where Rick talked about you might want to do something like to analyze across subjects, you might want to use a multi-layer analysis. This would be a way to do that. Subjects, it doesn't matter who's linked to who. So you would link each node to itself across all layers, okay? And so I guess I shouldn't really call these multitask networks, but these are categorical networks where you don't have a concept of time. So one way to think about this is just, if this is a temporal network, each layer is going to be a single functional connectivity matrix, okay? And I'm not going to go into all the problems of how we've defined what nodes are and what edges are. I'm going to assume that you have binged the rest of the day and you've already made those decisions. But now we're going to talk about, well, how do I actually then link these appropriately over time? All right, so because we have this different representation where we don't just have a single graph, we don't have a single matrix either, right? We're going to have different matrices that are kind of linked in some way, right? So the way that we tend to express this is through a supra-adjacency matrix. All right, so the way to think about this is you have each layer here, right? We have 3 nodes. And I should say that when I talk about this, when I say each node is coupled to itself, it doesn't have to be only coupled to itself. Okay, that is another reduction in complexity that people make, is to only connect a node to itself across time. But you can actually talk about how a node interacts with other nodes across time too. So in this case, we actually have node 2 linked not only to itself in the next layer, but also to node 1, showing that there's some sort of interaction between those 2 nodes that happened over time. Okay? And so when we put this in the superadjacency matrix, what we're going to have is essentially you just kind of stack your matrices. Okay? So the diagonal is going to be each of these layers. So the green here, this is, if we call this layer X and layer Y, this square here is going to be our X matrix. This one is going to be our Y matrix. And then this XY block up here, that's going to tell us about the interactions between X and Y, okay? Another way to think about this is basically nodes no longer have one feature that's defining them. It's not just node 1, it is node 1 in layer X. Okay, so another way to represent this would be to actually just use pairs to designate each node. It's node 1 in layer X, and then you can think of just building a matrix where each node has its own identity, but its identity has 2 pieces of information in it. Okay, and so this is going to be the superadjacency matrix. The nice thing is you can actually then work with the superadjacency matrix in many similar ways that you normally would with a normal adjacency matrix, and that's probably why this more simple form of a multi-layer network has been more studied, because the math is a little bit easier to do. You can look at this superadjacency matrix and you can do a lot of things. I should say that also, while this is another thing where the community is split, some people use these superadjacency matrices. You can also represent multi-layer networks with tensors. Okay, and so there's a whole camp of people that study these using a tensor representation too. And what paper you read, you'll get different types of notation for this. Okay, so that's your kind of overall introduction to multi-layer networks. So how do we actually build them? And I think temporal networks are probably the most studied type of multi-layer network, at least in neuroscience right now. And that makes sense because a lot of times what we're asking about is we have time series data, right? A lot of us here study functional networks, and we want to ask how basically the brain activity, how the interactions change over time. You want to look at learning, you want to look at development, you want to look at the progression of a disease, things like that. So you want to be able to say, well, if I look at this window of my data, what is the network structure and how does it compare over time and how does it change? Okay, so one way to do that would be to just build separate networks, right, and study them all separately. Okay, but I'm hopefully going to convince you by the end of this talk that if you instead build a multi-layer network, you have the ability to track things in ways that you can't do otherwise. Okay? All right, so if you're going to build a temporal network, basically what you're going to do is you're going to have some sort of time series data, okay? We can call each one of these from a brain region for simplicity. Your nodes are brain regions. And you're just going to choose some sort of windowing scheme, okay? You might choose it to be non-overlapping windows. You might choose it to be overlapping windows. A lot of what I talk about today is going to depend on your application, okay? So you're building a model, and how you choose to do your windowing, you just have to think about what is the interpretation of that. Do your time points overlap? Does that somehow smooth your network in some way? What does that actually mean? Okay, so you're going to window your data, and then you're just going to create functional connectivity matrices for each time window. Okay, and then you're going to take all of your matrices, align them all in time, and you're going to connect each node to itself in the following matrix. So this isn't particularly the best schematic of how to do this, because this is a functional connectivity matrix. So, but imagine that this is a node here, and you're just connecting to itself in each one, okay? So you have this temporal progression across your windows. All right, so what if we wanted to build a multitask network, okay? There's a lot of reasons we might want to ask how tasks are similar between themselves, or look at measures of communities between tasks, things like that. All right, so in that case, what we would do is we would separate our data into our tasks. And so instead of these temporal windows that are kind of sliding across time, now we're going to have our distinct windows associated with each task. And then we're going to use this categorical coupling. So we're going to take each node and we're going to couple it to itself in every matrix. Okay, so this is all-to-all between layer coupling. Another thing that people have looked at is, instead of task or temporal networks, frequency-based networks. Okay, maybe you actually want to ask how different frequencies are interacting with each other. So in this case, each layer of your network is going to represent a different frequency band. Okay, so maybe you just want to study the effects of different frequencies and you want to separate into frequency bands. Maybe you also are interested in studying the effects of cross-frequency coupling. Okay, so the multi-layer framework is very nice for that, because now you can actually, remember I said in most cases we only connect a node to itself in these layers? Well, you can connect a node to every other node across layers too, and you can get cross-frequency dependencies between your frequency bands. Right, so that's one application that people have looked at. So in this case, you're going to just decompose the signal into the frequency bands. Again, within each of those bands, calculate functional connectivity like you normally would. And then maybe you choose to calculate cross-frequency coupling. So in that case, what you're going to have, this would be your super-adjacency matrix over here. All right, so this is going to be the coupling within a single frequency band. Those are going to be these squares along the diagonal. And then these off-diagonal elements are giving you cross-frequency couplings. Okay, so that's kind of the basic overall of just building networks. Do people have questions about how to construct these types of networks from actual data? Oh, yeah. So the question is there a way to do it with structural connectivity? Absolutely. You can think about having structural connectivity, for example, across different subjects, okay? And so maybe you want to build something where each layer is a single subject, all right? And then you're going to ask, I'll talk about this in a second, one of the most common things that people look at in these networks is communities, because you can ask how communities evolve through your multi-layer network. And so you can ask about things that are similar between subjects, okay? So that would be one way of looking at it. Also, structural networks do change over time, okay? So you could think about trying to identify changes in structural networks if you have maybe longitudinal data where you're looking at some big developmental study and you have kids growing up or something like that. You could ask how the connectivity changes over time, even in a structural network. That is an excellent question, and that is something that I did not really discuss at all, and it's actually the most important thing, so I'm glad you asked that. Traditionally, what people do is they actually just choose a constant value, okay? And Oh, I'm sorry. So his question was, how do you determine how to couple a node to itself across these layers? Okay, and the answer is you're going to have this parameter, right? It's a connection and it has a strength. And I think that choosing the value of that strength between layers is actually the most under-researched and at the same time most important question when you're designing these. From a modeling standpoint, you really have to think about this. Let's say you have correlations or something. Well, let's go back to functional networks. And you have a functional network and you have correlations. So correlations, let's just assume they're all positive to be even more simple, okay? I'm a mathematician. We want to be as simple as possible. So you have something that's between 0 and 1 within a layer. Well, how do you choose the strength of coupling between layers? If you think about this, if you choose a value of 10, that coupling is so much stronger saying that a node is itself than anything within a layer. So you probably want to choose something that's between 0 and 1 to be like that. And so what we'll talk about when I start talking about community detection is this value. It's typically represented by omega in these interlayer couplings. And you have to determine how you're going to make that choice. And it turns out it actually matters. And doing community detection, just like in the single layer case, there's a multi-layer resolution limit. So you're not going to be able to detect communities depending on how you choose this parameter. So it's very much an unanswered question right now. So the best thing you can do is you can explore across a couple of different ranges, or probably the better way is to say that you have some sort of experimental data that tells you something about the similarity between nodes across time. Okay, this isn't going to be true about all types of data sets, but people in ecology are actually really good about this. When they build multi-layer networks in ecology, they tend to do their interlayer coupling based on observations of their data and how their data is changing over time. And that has been less employed in neuroscience. But I'm going to talk very briefly about some of the work that my group has been doing to try to think about that question of how do you actually determine how to couple these, and how would you figure out proper coupling strengths that are giving you meaningful information about going between these layers. All right, so that was a very long answer to that question. All right, any other questions before I move on? Okay. All right, so once you've built your network, you probably want to measure things about it, okay? So a lot of the normal network statistics that we've talked about today have actually been extended to multi-layer networks, okay? But again, there's this key question of how do you take this interlayer coupling into account? Often it's this artificial parameter that you've kind of put into your network, okay? So there are some methods that actually don't really even consider that. It's called agglomeration, where you're basically just going to collapse all of these layers onto itself, and you can think about doing this in different ways. So if you have these layers and you just collapse them, you can aggregate. You can also collapse across certain subsets of layers. So you can kind of say, well, these layers are more important than others. I'm going to give them different weights, or I'm only going to look at subsets of these and collapse them. And there are many different ways then to do these calculations. So unfortunately, I'm going to be even worse than Rick was in his I'm not going to give you any answers. Because the answer really is that you have to know what question you're asking about your data and figure out the appropriate way to do this analysis. Right? There's not going to be one easy answer where you can just look up a paper and say, oh, well, this measures degree in a multi-layer network. I'll use that. Because there's probably 10 different ways to do it, and they've all been defined slightly differently because of the properties of the data they were designed for. All right? So it gets a little bit more complicated when you start getting into this. Okay? So a good example of this is a clustering coefficient. All right? So this has been mentioned a few times today. A clustering coefficient is defined by counting the number of triangles within a network, and you're looking for how many triangles are present out of how many triangles could possibly be there. Okay? But now let's think about computing a triangle in a multi-layer network. We want to have the same notion of clustering, but how are you going to define a triangle? Because a triangle now can exist across different layers. Okay? This is even a problem in weighted networks. If you talk about weighted networks and trying to define a clustering coefficient, you have to ask how different strengths of edges correspond to kind of the strength of your triangle and what that represents. And people have come up with different measures for weighted networks. Again, it gets even worse when you go to the multi-layer setting, because now you have to ask, well, what happens if I have two edges in one layer and one edge is in a third layer? Or what if they're all scattered across different layers? How do I represent that? Are those equivalent triangles in my network? And again, I don't have a good answer for you, but I want you to be aware of the fact that this problem exists. So if you have some sort of multi-layer network that you've built, it's going to matter. Is it temporal? Are you looking across tasks? Are you looking across subjects? Because these interlayer couplings have meaning, and they're different depending on how you've designed your network. Okay, so how you would want to think specifically about triangles in your network is also going to relate to the system that you are actually modeling. Okay, and I'm sorry I'm not giving good answers, but these are problems that you need to be aware of, because this very much isn't a framework that you can kind of just, use as a black box and throw your data into and get it out. You really have to think about the choices that you've made when you built this multi-layer network before you go to analyze your data. If we think about random walks on a multi-layer network, we now have paths that can go through a single layer, but they can also move between layers, okay? And so this makes it really nice, because now you can get between nodes that were maybe, here we have two unconnected modules here, but in a multi-layer framework, there's actually a way to move between them, all right? You can jump into the next layer. But if we think about doing these walks on networks, again, if we're gonna turn this into some sort of transition matrix where we're looking at the probability of moving across an edge, the weight of that interlayer link is going to tell you how easy it is to move between those layers. So how you've made that choice and what that strength represents tells you something about how easy it is to go between the layers. So again, what you're going to want to do in most cases is, if you're just using this constant value across layers to represent links between layers, you're going to need to vary that value. This is very similar to the resolution limit in a single network case. You're going to have to actually look at the strength of your interlayer coupling and ask how changing that changes the properties of your network. Is it stable? Is it very sensitive to changes in that? Okay, so now I'm going to spend a larger portion of my talk talking a little bit about multi-layer modularity, because a lot of the other network statistics, like I was saying, are very hard to interpret at the end of the day. If we talk about clustering coefficient, what is a triangle in a multi-layer network, and is it measuring the same concept that we would associate with a single layer network? However, when we think about community detection and thinking about communities, this is something that is a little bit easier to interpret, okay? And so I think it's probably for that reason that this has been one of the more widely employed measures. So just like in the single layer case, we can extend modularity to a multi-layer case, all right? So this first half of the equation here is just your normal single layer modularity like Rick showed. However, what we add is this extra term here, and this omega here, I've said a few times, is the interlayer coupling, okay? So that's gonna be the strength of coupling between layers. And again, it doesn't have to be constant, okay? These JLR is actually telling you between, I think, here we go. It's between I've lost sense of what all these indices are. So it's going to be for node J, between layer R, whatever, time slice R, what is L? Slices, okay. So it's for node J between slices L and R, okay? And so basically, when you're looking at this, you can do modularity maximization in the same way that Rick described, but now what you're going to get is when you maximize the multi-layer modularity, you're going to get communities that exist across layers. Okay, so if we think about this where layers are time, what this means is that you have communities that are forming and dying and changing their membership over time. Okay, so this is a case of looking at, this is not an adjacency matrix now. Here we have nodes going down one axis, and this is layers on the bottom. So you can think about this as time, all right? And the color indicates the communities. So if you have this solid blue line here, that's going to tell you that this, I guess this is node 110 or something, up here, stayed in the same community over time, okay? But you can see that some communities, I think it's easier to look at this behavioral networks over here, where there are fewer communities. Some nodes change the community that they're in over time, all right? And some communities don't even exist at first. In the first layer of this network here, we have 3 communities, all right? The dark blue, light blue, and yellow. And then eventually, we see the emergence of a red community, okay? Something has changed about the network that a different community has formed. And so suddenly we go from having 3 communities to having 4 communities, okay? And then eventually this light blue one dies and we go back to having 3 communities, but they're different communities, okay? And that's one of the reasons that the multi-layer framework is so meaningful is that we actually can tell that this blue community started off but then died, and this red community started later but persisted. Okay, and so we actually know that there are 3 communities here and there are 3 communities here, but they're actually different communities. 2 of them are the same, but that third community actually represents something intrinsically different about our network. Okay, and that's actually one of the huge advantages of using this type of framework. The other thing that's nice is you can actually, now that we can define communities across time, we can use that fact to introduce new measures that we couldn't define before in just static networks. So we can talk about things like regional flexibility. So we can take a brain region and we can track its community allegiance over time, okay? And then we ask how many times does it change communities that it's in? And that's going to tell us its flexibility, okay? So it's just the number of times a region changes its module allegiance, the community it's in, normalized by the total number of changes it could have, all right? We can also talk about something called regional promiscuity. Okay, you can think about this. If a node is changing its community, it could just be jumping back and forth between 2 communities. Right? Maybe it's fluctuating between 2 communities. It's kind of on the border. Sometimes it gets lumped with one, sometimes the other. Or you could have some region that's actually floating around. If you have 10 communities, it goes and participates in all 10 of them. Okay? And those are very different types of interactions, all right? So you would not want to look at only the flexibility, perhaps. You might want to also ask about the promiscuity, because they're going to tell you what type of changes are occurring as a function of time. All right, you can also then use this fact that regions are changing their community over time to create what's called a module allegiance matrix. And this is just going to tell you what is the probability of 2 nodes being in the same community. Right? And you can think about this across a number of different dimensions. So you can look across all of your partitions. You could have something where you've run your algorithm multiple times because you need to do a consensus to see how many times regions are together. You also might have something where you've done this across a bunch of subjects. Okay, you can have different dimensions of your data. But if you look at a single partition of your community, what you're going to do is you're going to say, all right, let's look at region 1 and 2. Were they in the same community? Let's put a 1 there if yes, a 0 if no. And you're going to do that for every single community detection that you've done. And then what you're going to do is you're just going to average across your contingency tables, across all variables, or maybe only across one dimension. This is actually a choice you can make. And what you're going to get is you're going to get this matrix that tells you the probability of those 2 regions being connected. Okay? And if I have some time, I'll show you a brief reason why this is important in applications. But you can then use this module allegiance matrix to measure things like recruitment or integration. Right? So now we're looking at something where recruitment is going to give you the strength of interactions within a group. Now this assumes that you actually have predefined groups in your system. So you can think of this as cognitive systems, for example. You have the visual system and you have the motor system. So recruitment is going to tell you how often are the nodes within the visual system in the same community. What is, on average, do they have a high probability of being grouped together and being in the same community? And then integration is going to tell you, well, what is the probability that 2 nodes in different groups are clustered into the same community? Okay, so do you often get that a region in the visual system is in the same community as a region in the motor system? Okay, so these are 2 different types of ways you can look at this based on looking at this dynamic community detection. All right, finally, there's another measure that you can talk about, and this actually relates both static community detection and dynamic community detection. So I'm going to give one example of this, which is community stability. So the idea is first, you have some time series data, and you're just going to use the whole time series, and you're going to perform community detection. That's kind of going to give you the average communities across all of your data. Right? Then what you can do is you can use a multi-layer analysis to look at dynamic community structure. So you can take that time window, chunk it up into smaller windows, and run dynamic community detection. So the static community detection is going to give you kind of the average community structure, and then the dynamic community detection is going to tell you, well, how does that change within that over time? So then we calculated something that we called the community stability. So basically we said if you take one of these static communities and then calculate flexibility for each of the regions within that static community, how stable is that over time? So actually, stability is just 1 minus flexibility, right? Flexibility is going to be 1. If it's changing all the time, it's going to be 0. So if you want to think about stability, flip that and say 1 minus the average flexibility. And this is going to tell you how stable that community is over time. All right, so one more thing you might look at is we talked a little bit about this interlayer coupling and how it matters what that value is. And I said generally this term is held constant and it's going to be the same across all your layers. However, you can show that if you have some sort of data, this is kind of a schematic here where things are doing something and then there's a sudden state change in your system and everything changes, in this case everything suddenly becomes synchronized, you're not necessarily going to be able to detect that. It turns out there's also this resolution limit in multi-layer networks. And so what you might want to do is use some sort of algorithm to say if you know that there are state changes at certain time points in your data, you're actually going to need to lower that coupling term in between those layers so that you are actually sensitive to detecting these changes in community structure. And here's a reference to our work that goes through and gives an example of that. All right, I don't really have a ton of time to talk about applications, but I have uploaded all of my slides, so please feel free to go through them. And I know Sarah's going to talk about applications in the next talk, so hopefully she'll talk about some of these. But these are just examples of ways people have actually applied the measures I just talked about. But I want to take some time to end here and point out that if you go to my presentation online, I've tried to include some useful references because I don't have nearly enough time to really teach you about multi-layer networks and all the things you can do with them. But hopefully what you can do is go read some of these papers that will tell you more about this and just networks in general. We have a review article out there about multi-layer brain networks. There's some papers that have good references for some of the statistics. People have extended things like PageRank, eigenvector centrality, things like that have been extended to multi-layer networks, and these will be some good reviews. Community detection, again, has been extended to them. And then these are all the references for some of these newer measures that people have built based on community detection, and then this algorithm for trying to adjust your coupling if you know that you have state changes in your data. All right, so with that I'll end and hopefully have time for a few questions.
Any questions for Sarah from the floor? So one of the online questions was specifically related to software, and is there any specific software that beginners to the field can use to compute and analyze multi-layer networks?
So there is less software available for multi-layer networks, but it's becoming more and more available. I don't think that there's some, in the brain connectivity toolbox, there are some functions that will work for multi-layer networks. A lot of people are actually publishing code when they publish a paper, but you're going to have to go find their individual paper to get their code. GenLuvain, which is the popular community detection software, has been extended to multi-layer networks, and that's really nice for using that. That's in MATLAB. I should also say that InfoMap has been extended to multi-layer networks. Community detection actually is very popular amongst engineers. So if any of you here are comfortable looking through engineering journals and looking at things published in IEEE, there's a whole nother world of literature that exists where doing this multi-layer clustering is actually really, really popular. So there's a lot, and they tend to publish their code also. But again, you're going to get a lot of it being associated with a specific paper, and I don't think that right now I know of a good resource that has kind of collected a lot of the code together. But if someone knows, please do let me know, because I would love to know that too.
So perhaps one final question, once again an online question, and it relates to the distribution of edge weights. The question is, what does edge weight, PDF, heavy tail, power law, etc., affect community detection? Does that have an impact on community detection?
It does. So I don't know exactly what the relationship is going to be, but again, it's going to change how this interlayer coupling interacts with the intralayer coupling. And it actually gets really non-trivial to start looking at this. So this is one of the areas where mathematicians and physicists really need to spend a little more time looking at some of the fundamentals, because I don't think that we actually have a good intuition for what those relationships are, but it definitely will affect how the community detection performs. And this is true in the single layer case, by the way, also. depending on the actual distribution of your edge weights, and in fact the size of communities that you would expect and the distribution of community sizes, different algorithms will perform better or worse to find those. And so this is something where you kind of just have to play with some different algorithms and see, do you have stable community detection, do you not, and see how that affects it.
Thanks so much, Sarah. So please join me in thanking Sarah again for her talk. So our next speaker is Sarah Morgan from Cambridge University, and Sarah is speaking about applications of network neuroscience. Sarah.
So my name is Sarah Morgan. I'm a junior research fellow at Cambridge University, where I work with Ed Bullmore at the Cambridge Brain Mapping Unit. And I'm going to talk today about applications of network neuroscience in development, in health, and in disease, really drawing on some examples of work that's been done at the Brain Mapping Unit over the last couple of years to give you some illustrations of how these kind of analyses work in practice. So Stephen Hawking once said that the 21st century will be the century of complexity. And working in brain mapping and in neuroscience more generally, it's really easy to understand why that is. So what we're interested in, brain structure and brain function, actually really cuts across a huge range of different scales. And if we want to be able to say that we understand how the brain works, we need to understand not only what's going on at these individual scales, but also how they work together and how they interact. And so networks, as we've seen over the course of today, are a really powerful tool to try and understand this complex data. And for me, Olaf Spåns summed that up really nicely and sort of tried to really nicely explained why that is in Network Neuroscience in 2017. And what he said was that networks, firstly, they allow us to map and to model brain structure and function. So to be able to map out what connectivity looks like in the brain. They also allow us to bridge different scales of organization. And for me, actually, one of the most exciting things about networks is that on the one hand, they can be used to study something as small as a protein, if you're talking about protein structure networks, for example, where nodes are amino acids and you look at how they connect up together, right the way through to something sort of large, as macroscopic as social networks or food webs even. And when we're talking about the brain, which goes across so many different scales, having a tool like networks, which can be used at those different scales, is really helpful. And finally, networks also give us a really powerful framework to address some of the bigger questions. For example, what are the general organizational principles of the brain? So one way to do that, for example, is by using generative models, which were mentioned briefly earlier. For example, were used in this paper by Petr Vertec in 2012. And so a generative model is really a kind of simple algorithm or simple set of rules to generate a synthetic network. So for example, you might connect up nodes with a greater probability if they're closer together in terms of physical Euclidean distance. And so in this paper by Petra, she tried to generate networks with using some of these simple rules, for example, distance or how many neighbors two nodes have in common already, to create some networks and then to see how similar those networks are to the real brain structure. And what she found was that brain networks make this economic trade-off between the cost of their long-distance connections and the topological benefits of having those long-distance connections. So this is really just an illustration of how using networks can allow us to look at, to kind of play with the structure of these networks and to see what factors are important in determining the connectivity of brain networks. Of course, network neuroscience has become incredibly popular over the last 10, 20 years with a huge number of applications. And of course, it's not possible to cover even a tiny fraction of these in just half an hour. But what I'm going to try to do today is to focus on 3 particular examples of applications of network neuroscience to human brain mapping. And the examples I'll look at are in development, in health, and in trying to understand the variation between healthy brain networks, and then finally looking at disease and how can you compare brain networks from healthy controls and patients with a disease. So I'll start with looking at development. And of course, development's incredibly important because mental health disorders often emerge during development. And so if we want to really be able to understand what kind of goes wrong in some of these mental health disorders, then we need to understand what's going on during this crucial developmental period. In fact, 3/4 of all mental health problems are established by the age of 24. So this is a really vulnerable period for the brain. So what can brain networks tell us about development? I'm going to look particularly at a piece of work by Kirsty Whitaker and Petra Vertech, where they were interested in looking at brain structure. So these are structural networks. And they took structural MRI scans from 297 healthy volunteers aged from 14 to 24, so really over that adolescent period. And they started off by looking not at networks, but at the kind of underlying structural measures. And I think for me, one of the really important things to say about doing network analyses is it's really important to understand in the first place what the underlying metrics look like, possibly even before you go to the network stage. So Kirsty and Petra started by looking at measures, particularly at cortical thickness and MT as a putative measure of myelination. And they started by mapping cortical thickness and MT on the brain at 14 years. That's the first two rows. And then they also looked at change in cortical thickness and change in MT over that adolescent period. And so the first thing that they found was that cortical shrinkage in adolescence is coupled to increased myelination, which is this part H on the figure. So the change in MT and the change in cortical thickness were correlated, negatively correlated with each other. And in particular, regions with increased cortical thickness, which tend to be in these sort of association regions, in some frontal and temporal regions, were more likely to show, so those regions that had the decrease in cortical thickness were more likely to show an increase in MT over adolescence. So that's what's happening sort of at the level of just looking at CT and MT at regional level. What if we actually want to think about a network? And so the way that Kirsty and Petra decided to do that was to look at structural covariance networks. And the idea here is that you parcellate your brain up into a number of different regions. So, for example, you might have 300 different regions. You measure a measure that you're particularly interested in. So here that was cortical thickness. It might be something else. It might be volume, for example, or some kind of curvature measure, whatever you're interested in. But you measure the cortical thickness, or the other measure, at your 300 regions in each of your subjects. So you end up essentially with a matrix of the number of regions with the number of subjects and cortical thickness measured for all of them. And you take, so at a region X, for example, you now have cortical thickness measured for each of your, say, 100 subjects. And you can then correlate that vector with the cortical thickness measurements that you have for a different region, region Y. So you can correlate those cortical thickness measures between those two regions, and then do that for every possible pair of regions in the brain. And that gives you your structural covariance matrix. And so that tells you whether a cortical thickness or the other, whatever measure it is you're interested in a particular region tends to co-vary with cortical thickness in a different region across subjects. So that's how you calculate the structural covariance matrix. And Kirsty and Petra did that for their cohort of healthy adolescents, so the 297 healthy adolescents. From that, so that gives you a network. From that network, they then looked at regional degree. So they calculated regional degree measures for each of the regions, and what was interesting was they then compared those back to the changes in cortical thickness and the changes in MT, which I've shown you a couple of slides ago. And what they found was that regions with highest degree tended to be those regions where cortical thickness decreased most over adolescence, or the regions equivalently where MT increased most. And those regions were also the regions with the highest closeness. So that suggests that adolescent cortical consolidation, so this consolidation process of cortical shrinkage, seems to be topologically targeted towards the network hubs. The code for these analyses is all available on GitHub. So if you want to see how this kind of analysis works and to have a play with the code and the data yourself, then do go over to Kirsty's GitHub page, and you can clone or download the repository. And then she has this wrapper file which can take you through the different steps of the analysis. So this is a really great way of seeing how these kind of analyses are done. So it takes you through how to import the data, how to calculate the covariance matrices, so doing these correlations, and then how to calculate the nodal and regional graph theory measures and perform the statistical tests. And again, there you have file descriptions on the wiki, which are also quite helpful if you want to understand, or if you're trying to understand how different files fit together and what's what. Are there any questions about what I've said so far? Okay, I'll carry on then. So that's my first example. My second example of brain network analysis is so far what I've looked at is how networks for a whole cohort of subjects, but what can brain networks actually tell us about variability between healthy individuals? And the approach that I've talked about so far, the structural covariance approach, as I say, really works for a whole cohort of subjects. And so if you want to look actually at individual subjects and variability between individuals, really what you need is a way to get an individual network for each subject, or ideally. There are other ways around, but ideally you want an individual network for each subject. And so Jacob Seidlitz proposed a new method called morphometric similarity mapping to allow you to calculate an individual structural matrix for each subject, still based on structural measures. So you can do this, as I say, from structural measures from a T1 weighted scan, for example. Also, if you have them measures like MT, or diffusion weighted measures like MD or FA. And the idea here, the way that you calculate one of these morphometric similarity matrices, is you get a number of different measures for each region in the brain. So things like FA, MD, MT, regional volume, curvature measures, whatever you have really. You calculate those measures for each region in the brain, and then you correlate them between regions. So it's a pairwise correlation, and that gives you this morphometric similarity matrix. And as I say, what's great about this is it gives you an individual morphometric similarity matrix, so one per subject rather than one for the whole group. You can, after that, threshold and binarize the network if you want to, or not, or keep it as a weighted, unthresholded network. And Jacob showed that if you look at the properties of that network, it has modules in the same way that we might expect from other types of networks, DTI networks, resting state networks, etc. So you see the kind of the different modules. And the other really nice thing that Jacob showed was that morphometric similarity was correlated with connectivity from track tracing in the macaque. In other words, in the macaque monkey, where we have this kind of more ground truth track tracing data, if two regions had a high morphometric similarity, they were also more likely to be connected by a tract. So in that way, morphometric similarity can be used as a sort of proxy for anatomical connectivity. So Jacob calculated morphometric similarity matrices in that way for the same cohort of 297 healthy adolescents, and then looked to see whether or not the degree of those morphometric similarity networks, so you can calculate the degree of the networks as was described earlier, whether or not the degree of those networks was related to a subject's IQ. And what he found was that indeed it was. And he used a multivariate technique called PLS, partial least squares regression, to be able to link up the regional degree values with an individual's IQ. I won't go into that in a huge amount of detail. I'm very happy to talk about it afterwards if you'd like to. But essentially what that does is you have a degree value for each of the regions for every subject. And you also have an IQ value for each subject, or in this case, a non-verbal and also verbal IQ values. And the PLS looks for combinations of the degree, the regional degree values, which can explain the IQ of your subjects. And what Jacob found was that there were two PLS components, which between them explained about 40% of between-subject variance in IQ. And so the first PLS component was correlated with both verbal and non-verbal IQ, and really localized in these frontal temporal regions, particularly in the left hemisphere. And the second PLS component was more sort of localized in more visual areas of the brain, and correlated particularly with non-verbal IQ, but not with verbal IQ. So that's an example of how you can relate these kind of network measures, degree, for example, but you could also have taken some other network measure of interest to a kind of subject's behavioral score like IQ. So finally, what can brain networks tell us about disease? My own work focuses a lot on schizophrenia and on trying to understand what brain networks tell us about schizophrenia. Schizophrenia is a really great place to be looking as a network neuroscientist because schizophrenia is often described as a disease of brain disconnectivity. And actually, there's a huge amount in the literature already to suggest that connectivity, both structural and functional connectivity, is decreased in patients with schizophrenia compared to healthy control subjects. So what we were then interested in looking at was, having seen Jacob's great results relating morphometric similarity to IQ and explaining a lot of the variability in IQ, can you also use morphometric similarity to understand case control differences in schizophrenia? And does morphometric similarity and morphometric similarity networks show differences in schizophrenia? So the first thing that we did was just to look at morphometric similarity globally. So this is going back to what I said before about really understanding kind of the global trends in your data before looking particularly at networks. And what we found in the first place is that morphometric similarity decreases in patients with schizophrenia in 3 different case control studies. In Maastricht, one from Maastricht, one from Dublin, and the openly available COBRA dataset. So that's what's happening at the global level. But actually, that global decrease hides a regional pattern of case control differences. So if you look at this is a kind of weighted degree at the regional level, and then look for case control differences in that weighted degree, what we then found is that, again, in the 3 different data sets, we get a reproducible pattern of case control differences. And those were particularly localized, so particularly decreases in the frontal cortex and some temporal regions. And one of the things that tells us really interesting about this is if you actually look at the regional case control differences between patients with schizophrenia and healthy control subjects, they actually correlate with the morphometric similarity you see at a regional level in control subjects, in the healthy control subjects. And that's what this plot in part F is showing, that you get this negative correlation. In other words, regions which have high morphometric similarity in healthy control subjects tend to have reduced morphometric similarity in patients with schizophrenia. So that's sort of coming back to this idea of hubs, and again, seeing that the hubs of the morphometric similarity networks seem to be the ones where we see reduced connectivity or reduced morphometric similarity in patients. What we were then able to do was to try and link that up to gene expression from the Allen Brain Institute data. So the openly available gene expression data set, which gives you gene expression for a number of different points in the brain. And so we linked that gene expression up to our cortical parcellation so that we had gene expression for the 20,000 genes at each of the 300 brain regions that we were interested in, so the different brain regions where we'd seen these case control differences. And we were then interested to see, can we link these case control differences up to those gene expression data? And again, we use PLS to do that. And I won't go into the details, but what I really wanted to flag up was so we do that. We find that cortical pattern of case control differences, which I showed you before, is actually associated with expression of schizophrenia-related genes, which was really exciting. And to try to understand what the genes that were particularly enriched for our pattern of case-controlled cortical differences, what we then did was to calculate a protein-protein interaction network. And really the point that I want to make here is that even when we get away from the brain imaging data, and we've talked a lot today about brain networks based on MRI data and other types of brain imaging, but even when we go to a very different level, where we're looking at gene expression and proteins and trying to understand what changes in schizophrenia at the level of proteins and genes, we find another network. So we end up analyzing another network. And for us, the exciting thing about this network was that it has a very strong cluster of genes, which as I say, have already been related to schizophrenia in lots of the prior literature. Again, the code for this work is on GitHub. And all that remains is to acknowledge lots of the people who have done the work that I've been talking about today, particularly Kirsty, Jacob, Petra, and of course Ed. Thank you. Are there any questions?
In terms of morphometric similarity as a measure, you're incorporating data from diffusion MRI, structural MRI. So there's a lot of data that's being integrated into the same measure. How could we then use that to relate to a pathological mechanism? So if we see a reduction in structural covariance, how can we interpret that in a biological context? Does that, for example, can we relate that to something like demyelination or axonal disruption? What does that actually mean biologically?
Yeah, so I think first the thing to clarify is what morphometric similarity means. So when we're looking at similarity between two regions, morphometric similarity is quantifying the similarity between two regions in terms of their underlying measures. So when we see a reduction in morphometric similarity, in patients, what that means is that patients' brain regions are more dissimilar to each other than we expect in the healthy control subjects. In terms of how can we link that back to some kind of underlying biology, I think that's obviously a huge question and a big question for a lot of network analyses, not just morphometric similarity. And for us, this approach of trying to combine with gene expression data from the Allen Brain Institute has been really powerful in doing that, because you have the gene expression data, which tells you more about obviously what's happening at the underlying biological level. And so if you can look for case control differences in particular regions, and you see that those particular regions are enriched for certain genes, then, so for example, if they're enriched for genes linked to myelination, then that suggests that something is going on to do with related to myelination. So for us, this gene expression approach is a really powerful way of linking back.
Okay, thank you. Any questions off the floor for Sarah? So if not, I'd like to, well, first of all, I'd like to please join me in thanking Sarah for her great talk. And Alex and I would like to invite the speakers in this afternoon session up to the front, and we'll have a discussion. If there's any questions from the floor, we'll be happy to take those. And it will also go through some of the questions that have been posted online. So if the afternoon speakers want to come up, I don't think we have enough room, but perhaps some of us can gather around the... general questions related to any of the topics that were covered this afternoon? Yep.
Yeah, I have well, thank you all for your talks, brilliant, really. I have a question about the multi-layer networks, and I hope it makes sense. I was wondering if when you have a lot of time points, let's say, or a lot of experimental conditions, a lot of whatever you use as a layer, I'm wondering if this may bias your cross-layer detection of modules, because you're forcing into your super network a lot of artificial connections, so that the effect of the gamma is somewhat distorted, I think. I don't know if this is...
So basically, you have two parameters you're going to be looking at in this case. You're going to have the gamma and the omega. And so you're going to want to be varying gamma and omega, as it turns out. Because if you think about this, you have to think about kind of how the resolution limit is going, or the resolution parameter is going to act within a layer, but then you also have this coupling between layers. And so what we would actually recommend doing is doing a consensus sweep across a range of parameters. So what you can do is you can actually think of this as kind of one extra dimension you have to run in your clustering, is that you actually have to do the community detection as a function of both different gamma and both different omega. So you're going to have some sort of matrix, essentially, of parameters, and you're going to run the multi-layer detection, and then you're going to do a consensus analysis across all of those different partitions.
So I think that the answer is potentially yes.
Yes, yes, it definitely will. But again, we don't really understand the interaction between them. And so there's some algorithms out, there's this CHAMP algorithm that tries to optimize how you would choose gamma and omega based on kind of the principles of how they are expected to interact. But we've found, actually, if you just do this sweep across different parameters, basically what you're going to be doing by doing that is essentially looking for some stable range. Because what's going to come out is that if you have a range that's stable over some range of gamma and stable over some range of this omega interlayer coupling, that's going to give you similar community detection across all of those. And so then when you do the consensus across all of those sweeps, what you're going to get is the stable partitioning. And so that is my recommendation about how to do this. But I think, again, that's a very open question about how you properly figure out that interaction.
Thanks.
Did you want to come up to the microphone so that we can... There's a microphone just to the left of you.
First of all, I have to say apologies if this has been answered in the morning session, because I was in a different educational course. But my question is about if you have acquired high resolution data, so you haven't covered the whole brain, are you allowed to do graph analysis? Are you allowed to define nodes and do network analysis if you're missing some part of the brain?
Yes. But you should be careful if you're talking about something that, like a network measurement that deals with the rest of the brain. So for example, like shortest paths, for instance. You may, if you're looking at a restricted subset of regions and you compute the shortest path between every set of regions or every voxel in your restricted subset of areas, you're necessarily missing out on paths that could potentially be shorter, but that go outside the area you're looking. So of course you can do this. Of course you can do graph analysis on a smaller subset of a restricted part of the brain, but you'd potentially miss out on something like the shortest paths that involve other areas.
Sorry, may I have a follow-up on this? So I assume that relates mostly on, so you compute some nodal metrics, but can you still do community detection? Okay, thank you.
People do analysis of, for example, ECOG networks all the time, right? Where you're only recording from where those electrodes are located. So that's something to keep in mind. You just always have to think about how you are interpreting your results, all right? Like everything we've talked about today, it's the interpretation that matters. So you just have to remember what you started with.
Any questions from the audience? Any further questions? Yep.
I have a question about the community detection. So for the community detection, if we put it in the time series, so I think one issue of community detection is about the change of the nodes assignment. Another one is the connectivity within or between the communities. I want to ask, are there any other aspects we should consider about the community detection?
It's probably Rick or Sarah.
So are there other issues to consider besides, what was the first one again? It was...
Oh, yeah, in my project, I consider the community detection problem includes, one aspect is the change of the nodes assignments. So
across time.
Yeah, across time. So how the nodes are allocated into different communities. So this is one aspect that I'm considering. Another one is the connectivity between the communities or within the communities. These are the second issue that I'm considering. Are there any other aspects of community detection that we should also consider it's changed or not in the time course?
I think a lot of the measurements that Sarah proposed at the end about characterizing multi-layer, so she talked about community detection in the context of multi-layer networks that describe time-evolving networks, temporal networks. All of the promiscuity, flexibility measurements are other measurements that you could probably consider, if I'm understanding your question correctly.
Okay, another question is, for the community detection, if only one nodes are merged into new community, or in a specific community, one of the nodes are split out, can this considered as a community detection problem, or is it precise enough to detect these kinds of?
So it depends on the algorithm you're using, but that's exactly what I was talking about when I was talking about this multi-layer resolution limit, is that actually you run into this problem of if you have small communities that merge or split, you can't necessarily detect that. We have a paper on arXiv that goes through some examples of that, and we're in the process of trying to publish a more full paper that kind of goes through some of the specific examples of trying to figure out when that's going to be a problem. You can get some bounds on when you can and cannot detect that. The resolution limit that I'm describing is particular to modularity maximization. So you can look at some of the other algorithms that people have proposed out there. Again, the engineering community is actually really into this right now, and so there's a lot of things that are based on like ICA and PCA and things like that across multiple dimensions that people are looking at. And so maybe some of those algorithms might be more useful if you know that that's going to be a problem in your data. When we were doing this, one of the things that I skipped over in my slides is if you look at networks of individual neurons, and you look at their firing properties, you tend to get a lot of things that are isolated. So you'll have single neurons coming together, and we definitely see that modularity maximization has problems getting some of those changes over time, depending on that specific choice of interlayer coupling that you use. So it is very much a problem. And again, there's an archive paper about it, and hopefully there'll be a real paper shortly.
Okay, thank you very much.
So I might take one of the online questions. I think this probably relates to Martin's talk, and it's specifically in regards to Rich Club and comparing Rich Club measures in the case where each individual has their own specific null. So is it feasible and does it make sense to compare Rich Club metrics when each individual has their own specific null model, a tailored null model in other words?
I think I'm going to do a Rick one here, saying it's really difficult to do this. It's a really hard problem. No, but I think it's a really good problem. Well, it's a good question because I think we all have to deal with this. So if I translate the question a little bit, let's say if you want to compare risk loop coefficient, or I think it holds for any type of other graph metric, then should I use my my own subject as some sort of a null model, right? Should I first create normalized metrics and then compare across groups, or should I come up with some sort of a general null model? I think here too there is no one single answer to this, to be honest. I think I would be more intuitively go for the first one, to first compute normalized metrics within a subject and then compare across subjects. But here too there are quite a number of limitations. Network density, for example, being the most simple one. If you have a higher level of network density, then also your null model will be affected by this. So although we mostly tend to use the null model to take out these effects of density, they might also introduce. And I think clustering coefficient is a very good example for this. If you have MATLAB on, then you can actually try this out, that higher random density networks also tend to have a higher clustering coefficient themselves. So in the funny case, you can actually have no effects between your case and controls, but when you normalize, you do see differences between cases and controls. So here too, in my opinion, there is no simple answer to this, but if any one of you have a good answer, then go for it.
Thanks, Martin. Any final questions from the floor? If not, I think we can wrap things up. I'd just once again like to thank my co-organizer, Alex Fornito, who's done all the heavy lifting in organising speakers and organising the workshop, not only this year's workshop, but also over the past few years when we've run this at the other OHBMs. Once again, please join me in thanking our speakers this afternoon. And I'd also like to invite you, if you have any comments on the workshop or any suggestions on how we can improve the workshop in future years, please feel free to come and speak to either Alex or I after the workshop, or send us through feedback via email. We'd be very glad to hear your thoughts on how we can improve the workshop and how we've gone in terms of organising it this year. So I think that will wrap up the workshop. And thanks again for your attention, and thanks for attending.